So, hello students, welcome to Vedantu NEET English. This is Baswaraj sir, your biology master teacher students. Good morning, each and every one of you students. Good morning. Now, all of you might be aware that what is your mock score range. If you have written any of the previous mock tests from Vedantu, obviously from AITS, everything, you will have some general idea that what is your current mock score. Yes or no, students? Yes or no? Please, students in chat. You will know a current mock score, your current score. You will have some idea about it right now, right? Now, students, I want all of you. Obviously, if you are scoring anything between 300 to 400 or 400 to 500 or 500 plus above, all of you wish one thing. Sir, how can I jump from the current mock score to 600 plus? Tell me, students, yes or no in the chat? Yes or no in the chat for all of you? So your current goal has to be right now to jump above 600 plus if you want a government medical college. So all of you need to be aiming at 600 plus, especially if you want a good college. 640 plus should be your aim right now, students. Should be your aim right now. Now, students, can everyone in the chat tell me what is your current mock score, students? All of you in the chat, students. What is your current mock score? All of you. What is your current mock score, students? In the chat, quickly, students, quickly. Students, all of you in the chat, what is your current mock score? Students, again, I'm telling you in general, right? At this point, if you're studying throughout the year, if your mock score is around 600 plus, right? That's a very good thing. If it's around 500 plus, yes, there is still room for improvement with the help of question practice, right? Now, students, again, in this particular session today, the entire session, all the way from your biology, botany, zoology, physics, chemistry, you will have a general idea that how many mark score you can jump. Okay, so especially students in biology today right now, my entire session is divided into 3 parts. The first part is called as your easy direct questions. The first part is going to be easy direct questions. Here, all of you will be able to solve these questions very easily. The second part is going to be the tricky questions. This is the region, friends. Let me tell you again. If you completely master the tricky questions, you are set. Because this part 2 of this video today only, the second part of this today's session, right, you will realize where you are losing marks, in general. The third part is going to be AR questions. Again, all of you need to really work on the AR questions because this is the part where you will lose marks again. So today's session is modified. It is curated in such a way that you can do the jump very easily, students. And again, I'm telling you, students, these questions are direct questions. Yes, in your examination, you will have some direct questions. Then you will have some tricky questions and AR questions. So if you master today's session properly, I can completely say, students, you will have a proper jump in your marks, students. So all of you in the chat, students, are you ready for today's class? So can I get some fire emoji in the chat to let me know, yes sir, we are ready to completely kill today's class. And yes, students, if you're new to the channel, quickly subscribe to the channel. If you're new to the video, quickly like the video, students. Students, all of you ready? Students, all of you ready, students? Quickly in the chat, tell me in the chat. It's fluctuating a lot. Yes. All of you students. Shall we start? All of you in the chat. Can I get some fire emoji in the chat, students? Can I get a fire emoji in the chat to start? Yes, sir. Let's go. Part 1, part 2, part 3. Starting right now, students. Starting right now. The first question on the board. Here we go, students. Again, these are fairly easy questions. All of you can solve these questions very easily. And these are the questions which will give you the borderline, the intermediate marks, that 100, 200, 300 marks are there, right? The easy 300 marks can be obtained by these questions easily, students. The first question on the board. Select the correctly written scientific name. Again, sometimes what they will give you, they will not give you the example of Mangifera indica. You will need to identify which is the correct scientific name here. Okay, students, is it option number 4, option number 3, option number 2, option number 1, students? See, again, this is not going to be the right answer. Obviously, this is not going to be the right answer because here everything is capital. Here you can see small letter, so not going to be the answer. Here you can see, students, Mangifera indica, but it is written not italics. It is written in a proper order, so it's not going to be the answer. So option number 3 is right, students. Option number 3 is right answer. Why? Because Ficus bengalensis, capital, small, italics. Done. Proper answer, students. Proper answer. Now that is an easy question. So there are certain easy questions, certain difficult questions. It depends upon you which is easy for you, because if you have revised the particular topic, it will be easy for you, students. It will be easy for you. Now, next question on the board. Which of the following are colonial green algae? This right here is a NEET PYQ also. Right? Chlamydomonas is going to be single cell. Volvox is going to be a colonial. Spirogyra filamentous, right? So Volvox is going to be a filamentous algae, students. Volvox is going to be filamentous algae. Yes. Students, done? Done? It's going to be Volvox is going to be filamentous algae. Students, again, I'm telling you in my chat, in my class, in our Vedantu NEET English, we always tell you to participate in the class because, students, it is okay to make mistakes. The more mistakes you make right now, the less mistakes you'll make in examination. The simple rule of our channel, right? So everyone should participate in the class. Everyone should attempt the questions. Everyone should do it. Then only you will learn. Without participation, you will never learn, right? Students, next question on the board. Here we go. The sugarcane plant has dumbbell-shaped guard cells, taproot system, reticular venation, open vascular bundles. Okay. Students, first of all of you should know, students, here sugarcane plant is monocot or dicot? All of you know, students, monocot is dicot or dicot or monocot? Monocot or dicot? Students, you will tell me sugarcane is a monocot. Sugarcane is a monocot because it belongs to grass family. Belongs to grass family now, students. Okay. So it cannot have taproot, students. It cannot have reticular venation because reticular venation is exclusive to your dicots here. Open, no. In the case of monocots, it is going to be closed because the cambium is absent. All of you should be aware that in the case of monocots, the cambium is closed here. Cambium is absent. So it is going to be closed here. So in the case of sugarcane, we also know that we have dumbbell-shaped guard cells. Dumbbell-shaped guard cells are found in the case of sugarcane. In the case of sugarcane, dumbbell-shaped guard cells are present. Okay, students. So 1 is the right answer here. Let's go, students. Let's go next question on the board. Oxosomes or F0, F1 particles, which are also called as what, students? They're also called as ATP synthase. ATP synthase, right here, particles occur on. In which region do you occur on, students? Students, I remember all of you, don't worry. I remember all of you, students. Okay. So F0 exosomes or F0 F1 particles or ATP synthase occur on, students. Outer mitochondrial membrane, obviously not outer, nothing is there, it's more permeable nature. Matrix, no. Peri-mitochondrial space, inner mitochondrial membrane, students. Now again, all of you should be aware that, students, here the best way to remember it is, remember, students, where does the ETC takes place, because the F0 F1 particles are part of ETC, electron transport chain. We have complex 1, complex 2, complex 4. So when 1 to 4 complexes, this ATP synthase is actually the complex 5. All of you remember, it is actually the complex 5. Students, quickly tell me right now, where does the entire ETC takes place? Students, you will tell me, students, entire electron transport chain takes place in the inner mitochondrial membrane. All of you, yes or no? Yes or no? It takes place in the inner mitochondrial membrane, entire ETC. So this again is part of ETC. So what is the answer, students? What is the answer? Answer is going to be option number 4 right here. So do not get confused based on what you're asking you. So always remember, oxosomes or as your F0 F1 particle, it is also called as your ATP synthase, also called as complex 5, which is a part of ETC. Done, students? Done, all of you? Done? Yes, students. Quickly smash the like button, all of you. First of all of you all, quickly smash the like button right now. I can just see 167. What is this? Quickly make it 500. Quickly make it 500, students. Next question on the board. The centromere splits during dash stage of meiosis. Now again, they're asking you meiosis right now. So you should be very clever. You should be very clever and tell me, students, do you think the centromere splits in your, do you think it completely splits in the anaphase 1? Or anaphase 2. See, these are two options you might get confused with. These two options are very easy. Again, we can remove them directly, right? So if all of you remember, students, all of you remember that I have told you very clearly. In the case of anaphase 1, what happens? In the case of anaphase 1, what happens is segregation of homologous chromosomes happens. What happens, students? Separation of homologous chromosomes takes place here. Homologous chromosome separate here. Homologous chromosome separate here. But the centromere split happens where? The centromere split happens only in the case of anaphase 2. Now, students, if I change the question to mitosis, and I remove all this, I remove one, then I'll change this to telophase. I'll change this to telophase. Then what is the answer, students? Then what is the answer? In such a condition, what is the answer, students? In such a condition, what is the answer? For example, centromere splits during dash stage of mitosis, anaphase, metaphase. Here, let's say I'll add your PMAT. Prophase I will add here. Prophase here and telophase here. Then also, students, then also the answer is going to be what? Then also answer is going to be anaphase itself. Clear all of you? Clear all of you? It's going to be anaphase itself. So please be clear on what they're asking you. Next question on the board. Glycerol is also known as, what is the other name of glycerol, students? What is the other name of glycerol? Quickly in the chat for all of you. Trihydroxypropane, dihydroxy, tetrahydroxy, monohydroxy. See, all of you remember, students, in the case of your glycerols, right? It has 3 OH groups. It has 3 OH groups, students. Okay. Yeah, save the chat, huh? Save the chat. Yeah. Students, what is the answer now, students? What is the other name of glycerol, all of you? Again, these are direct questions. It doesn't require a lot of thinking. You can answer these questions without even, spending more than 10 to 20 seconds on the question, right? Trihydroxypropane. Very good, students. Next question on the board. Here we go. What is the correct order of stages of cellular respiration? Now they're asking you, what is the stage 1 by 1 for your entire cellular respiration? Okay. First, do we have Krebs cycle? No, absolutely not. See, these two options are gone because glycolysis does not come in the end. Glycolysis always comes in the first here. Okay. So glycolysis, Krebs cycle, electron transport system here. Option 1 is the right answer. Okay. We cannot have electron transport chain coming first. Electron transport chain has to come at the very end here. Okay. So here is the answer, students. Now again, students, can anyone tell me between the glycolysis and the Krebs cycle, which is the stage coming here right now? Between the Krebs cycle and the glycolysis, there is one more stage in the middle, right? Before Krebs cycle and after glycolysis. That is called as what? Link reaction. Link reaction is missing, also called as your oxidative decarboxylation reaction, also called as gateway reaction is missing here. Link reaction is missing here. Okay, so remember that much also, all of you students. C for what, students? C4 in the chat, all of you. C for cat. What even? Okay, next question on the board, students. Heterophilous development due to the environment can be an example of. For example, all of you have a certain life cycle, right? Now any plant deviating from the life cycle, which can result in different shape of leaf or different type of leaf in the case of plants. What do you call it, students? What do you call that? What do you call it, students? What do you call that? Change in the life cycle is because of change in the life cycle. For example, in the case of buttercup, larkspur and cotton and coriander. In the case of buttercup, larkspur, coriander, cotton, a certain types of life cycle is called as plasticity, students. It's called as plasticity. Okay, plasticity, students. Next question on the board. Dash is used to speed up the malting process in the brewing industry. Again, this is a previous NEET PYQ. Again, NEET PYQ. And again, students, the previous question also was a NEET PYQ. The previous question also was a NEET PYQ, students. Again, what is the answer? Again, students, what is the answer? Dash is used to speed up the malting process in the brewing industry. NAA, no, it's an oxen, no. GA3 is gibberellins. All of you should know GA3. Yes, GA3, students, exactly. GA3 is a gibberellin which is used in your malting process. Now, again, students, if I change the question to, let's say, increase the grape stalk length, what is the answer? If I ask you the question, increase the grape stalk length, then answer is going to remain the same. If I ask you, students, increase the internodal length in your rice, in the case of your sugarcane, same answer again, right? Which of the following hormone is used to spray on the juvenile conifers? Again, same answer, students. Okay. Now, students, if I change the question to, let's say, immediate closure of stomata, then what is the answer? Which of the following hormones involved in the immediate closure of stomata during your unfavorable conditions or harsh conditions? Which is the hormone involved there? Okay, then answer is going to be what? Then answer is going to be abscisic acid. In such a case, abscisic acid is going to be the answer then. Next question on the board, students. A type of population interaction in between two population of two different species where one species benefited and the other neither benefited nor anything else. For example, I am benefiting, you are benefiting from, right? All of students. Yes. All of you students. Then what is the answer, students? What is the answer? Where one is benefited, other one is neither benefited nor, it doesn't care. It doesn't care, students. It doesn't care. That is going to be commensalism, students. Can anyone give me 3 examples of commensalism? 3 different examples of commensalism. One is orchid and mango tree, sea anemone and clownfish, sea egret and the buffalo or the cow. I need one more example of commensalism, students. I need one more example of commensalism in the chat. Examples of commensalism. Now again, if you can tell the examples of commensalism, students, can anyone give me an example of amensalism? Let's say. Again, it is not given in NCERT. Just check your knowledge here. Just to check your knowledge here. Can anyone give me an example of amensalism also? Okay. Now again, students, next question on the board. Dash proposed the Rivet-Popper hypothesis. Who proposed the Rivet-Popper hypothesis, students? Who proposed the Rivet-Popper hypothesis? Again, students, in the chat quickly. Who proposed the Rivet-Popper hypothesis? Yes, all of you students. Barnacles on whales, absolutely, students. Barnacles on whales, absolutely. Antibiotics, exactly. Antibiotics, fungus like bacteria growing, right? That is an example of amensalism. Black walnut is an example of amensalism. Yes, Paul Ehrlich, right answer, students. Paul Ehrlich. Now, can anyone tell me, students, in Rivet-Popper hypothesis, what is who is the ecosystem here? Who is the ecosystem here and who is the species here? For example, in Rivet-Popper hypothesis, Paul Ehrlich told us that this entire structure is the ecosystem, and in this ecosystem, certain small parts are called as species. So, what are the species here and what are the ecosystem here? What is the ecosystem here? In the chat, all of you. Now, next question on the board, students. These are regarded as the major cause of biodiversity loss. Now, here they have given you multiple options. You're supposed to tell me out of these multiple options, which are the part of evil quadrant, students. Part of evil quadrant, students. Part of evil quadrant. All of you in the chat. Aeroplane as a, yes, ecosystem. Species as your rivet or screws. Beautiful, beautiful, students. Beautiful. Yes. Animal destroyed by the feet of elephant. Yeah, true. That is also amensalism. Correct, students. Shamla, Meghana, Naina, Yash, Chintu. Chintu, nice name, bro. Meghana, correct. Ashley, correct. All of you. Right answer, students. Now if I change the question to, these are regarded, I'll change it to, I'll change the question to, students, this much. The major cause of biodiversity loss. Out of all the following, students, out of all the following, if I change the question to major cause of biodiversity loss or the highest reason, the most effective reason for biodiversity loss, the answer is, students, then the answer is going to be habitat loss and fragmentation. In such a case, students, habitat loss and fragmentation is the answer in such a case. Okay. Yes. Now, students, again, this is the last question of your easy questions. Okay. This is the last, this is the last question of your easy questions. After this, some tricky questions are going to start, students. Last question of your easy quill. Last question of your easy quill, students. Last question of your easy quill. Robert May, students, again, see, right now, Paul Ehrlich is Rivet-Popper hypothesis. Okay. Alexander Humboldt is going to be your species-A relationship. Robert May is going to be your global diversity of 7 million. Then David Tillman is going to be your long-term ecosystem experiment using outer plants. So what is the answer, students? What is the answer? What is the answer, students? Again, option number 1 is the answer, students. Now, students, all of you ready now? Are you ready for certain tricky questions right now? Certain types of tricky questions. Now, these are easy questions, students. Now, again, I want all of you to rate your question efficiency solving of your easy questions out of 10. So, no, out of 5. How easy was the easy questions out of 5, students? How easy was the easy questions out of 5? All of you in the chat. How easy was the easy questions out of 5, students? Quickly in the chat. Yes? All of you in the chat. How easy was easy questions? Easy, very easy, or too easy, or little difficulty, students? Yes? All of you? 5 out of 5? 4.5 out of 5? Now, I believe, students, easy questions, I don't think so anyone would go below 4 at this point. Easy questions, I don't think so anyone would go below 4. So I believe I want all of you to keep the same enthusiasm, same energy for these questions also. So students, let's go with the next questions right now. Nice. Now here is the first question on the board. Take your time. I'll give you 30 seconds and solve the question and tell me, students. Take 30 seconds and tell me the answer, students. Again, 30 seconds. Let's go. Fall has two pure forms, black and white. If black form capital B and the white form small capital W, white form are crossed, the F1 individual appear to be blue colored, capital W, due to the incomplete dominance. Again, students, incomplete dominance. Now this is because of incomplete dominance. So as soon as you see incomplete dominance, your answer is supposed to be option number A. If you do that, students, you are wrong here. If you put option number A, as soon as you see incomplete dominance, your answer is going to be wrong here. Now I'll tell you, because this is where the tricky question they catch hold of you. Which of the following would be the outcome of cross between black form and blue form? Now again, students, if you go to option number 1, all of you students in the chat, if you put option number 1, I want all of you to put red hearts in the chat if you put option number 1. I want everyone to put red hearts in the chat if you put option number 1, because I want to know how many of you got it wrong. Okay. Now they're asking you which of the following would be the outcome of the cross between black form and blue form. Now again, students, black form is what? Black form is capital W, so capital B. Blue form is what? Blue form is capital B, small capital W. Let's do a cross. Here, what are the gametes we get? We get capital B here. Gametes here, what do we get? These two gametes. Do a cross here. Let's do a cross here. What do we get? This is what we get. This is going to be black form. This is going to be what? Blue form. Now 1:1 ratio. So we get one black and one blue. We get one black and one blue, students. So answer is what, students? The answer is going to be one black and one blue. So these are called as tricky questions because they trap you in the starting. Did you also get trapped, students? Did you get trapped? Because as soon as you see incomplete dominance, your mind is tuned to look at this answer, and that is why this option is given at first. Why do you think this is given at first? It is given at first to trick your brain thinking that incomplete dominance. So first answer. So but you should be very smart in reading the question what you're asking you, students. Now students, now tell me, all of you, are you ready for tricky questions now? Friends, all of you, are you ready for tricky questions right now? Because yes, these questions you want to look at twice. You have to look at them at twice. You don't have to look at once, because if you look at once, your brain will be tricked into going wrong answer, students. So students, ready all of you? Ready all of you for tricky questions right now? This was the trailer. This was the trailer here. All of you ready now for the full movie right now? Ready? All of you? Yes? Yes? So I want all of you to concentrate right now. Again, these questions are different little. Okay. Now, students, next question on the board. Here we go. Identify the correct sequence of taxonomical category of mango. Now, again, students, all of you might be aware of see, I'll ask you usually what they ask you. They ask you family or genus or order. They ask you, but here you need to know the entire classification of your mango. You need to know all the way from the scientific name to your order and the family, everything. So, what is the answer, students? What is the answer? Mangifera indica. And it says taxonomical category also. It has to go in order also. Students. It has to go in order. You have to know correct option also. Good question, no? Good question. These are called as the tricky questions which will decrease your marks in general. These are the questions that are designed to decrease your marks. So what is the answer? What is the answer, students? What is the answer? Now, students, see here, what actually comes first? In Mangifera indica, always go from Mangifera, Anacardiaceae family comes first, order comes, then class comes. Now, students, tell me the answer. Family, order, all of you know. Tell me the answer, student. Tell me the answer now. What comes first? First, scientific name, then we have the family, then we have the order, then we have the class here. So what is the students? Option number D is right answer. Some of you might have gone wrong because thinking that option number C is right. You might have written Sapindales first, then Anacardiaceae. No. What has to come first? Anacardiaceae, the family has to come first, and then we have to get Sapindales. Then we have the entire entire class here, students. Now again, if I ask you questions, if I ask you a question if I ask you a question from any of the following, will you be able to tell? Will you be able to tell for your, let's say for housefly, what is the entire taxonomic category? For humans, you can easily tell. For wheat also should be able to tell. Triticum, Poaceae, Poales, Monocot. So you should be able to tell, students. You should be able to tell. So next question on the board. Here we go, students. Here we go. Tricky question. Take your time. Do not answer in a hurry to be the first person. It's better to be right than the first person. It's better to be right. Question on the board. Here we go. Question on the board, students. Here we go, students. All of you. Students, have you liked the video? Because students, all these questions takes time. So I want all of you to quickly smash the like button also. Choose the incorrect. They're asking you to choose the incorrect statement here. Choose the incorrect statement given below. In 1892, Ivanovsky identified the microbes as the cause of tobacco mosaic disease, smaller than the bacteria and able to pass through the filter. Okay. Diner later showed the extract from the infected tobacco plant could infect the healthy ones, coined the term virus. Benchenic work in 1935 crystalline viruses ever revealed their protein composition and obligate parasitic nature. So what is the answer? What is the answer, students? Take your time, take your time. Do not, it's better to be right than, lose your marks here. See, all of you should know by now, students. Here, I'll give you a small recap here, students. See, first one, Ivanovsky. Yes, he worked on your microbes causing your tobacco mosaic. Yes, for your TMV. These were found to be smaller than the bacteria because they passed through the filter. So, first statement is right here. First statement is right. Now, students, Benchenic in 1898 demonstrated that extract from your infected plant tobacco could cause the infect in the healthy plant. So, students, here it says diner here. So this statement is wrong here. This statement is wrong here because it was Benchenic here. Now, last statement you see, students. Benchenic work it says, right? But all of you should know, students, it was your Stanley showed the virus could be crystalline. So it was Stanley, not Benchenic. So this statement is also wrong here, students. So what is the answer, students? What is the answer? Which of the following are wrong statement? 2 and 3 are going to be your wrong statement, students. 2 and 3 are going to be wrong statements, students. So all of you should know NCERT lines can come like this in your examination. Next question on the board. Here we go. Take your time. This is an easy question actually. This one is a fairly easy question, right? This one is an easy, fairly question. Students, quickly smash the like button, all of you. And students, I want every one of you to call your friends, tell them, students, yes, there's a channel which are doing not only easy questions, they're doing tricky questions to make you, understand the concept here at this point also. All of you. Protonema stage and leafy stage, this is an easy question, doesn't require much thinking. Okay, this question you see, student, this question you see. Now here, you need to identify what is the estivation here. Estivation here, and after identifying the estivation from the diagram here, you should also know the example of this estivation. It's not very just important to even to know the estivation, you need to know the example of estivation here. So all of you should know, students, this one is completely on the inside, this is completely inside here, this is completely outside here, inside, outside, and remaining 1, 2, 3 are alternating with each other. So can I call it as imbricate estivation? Yes, imbricate estivation, students. Okay. Imbricate estivation. So if it is imbricate estivation, it's not twisted, it's not twisted. It is imbricate. It is twisted is supposed to be alternating. Imbricate, imbricate, advocate. Advocate fights gun cases. Gulmohar and Cassia. Gulmohar and Cassia is the answer, students. Gulmohar and Cassia is the answer. So option number A. Again, it is not twisted again. It is imbricate because one is completely inside, one is completely outside. Remaining are alternating with each other, right? So this is a imbricate estivation. If this was outside, then it would have been twisted here. Okay, so Gulmohar and Cassia are the answer, students. How are the questions? Students, how are the questions? These questions will make you think. You need to identify the estivation, then you need to know the estivation, then you have to know the example here. So just because if you know the diagram, you will not get the marks. Just because you know the estivation, you will not get marks. You need to even know the example also. These are called as tricky questions again, students. Now, next question on the board. Next question on the board. right? Which among the following shows the correct position of the floral part of thalamus in cucumber? Again, you need to know cucumber belongs to which type of your ear. Then you know the diagram also. You need to know the diagram also. Then you can get the right answer to again. Cucumber, ray flowers of sunflower, all of them belong to one category. Cucumber, right? Ray flowers of sunflower, all of them belong to one category, right? All of them belong to epigynous condition, right? And epigynous condition is only one option here. That is going to be, students, option number C. Now, students, I'll change the question to let's say I'll change the question to, let's say, brinjal. I will change the question to brinjal. Then what is the answer, students? Then what is the answer? If I change the question to brinjal, then what is the answer, students? If I change the question to brinjal, then the answer is going to be option number D, because superior ovary here, superior ovary. Both of them are perigynous condition. Both of them are perigynous condition, students. Done? Done. Now, next question on the board, it's a simple match the following. Doesn't require much brain cells. All you need to know is remember the mnemonic, start matching. Okay. Marginal placentation, pea plant. Marginal placentation, what? Pea plant. Axial, allow China to leave. Tomato, lemon, and axial placentation. Parallel parents are mustard, argemone and mustard. Argemone and mustard. Free central, students, dianthus and primrose. Dianthus and primrose. So what is the answer, students? What is the answer? Option number, students, B here. Option number B. Next question on the board, friends. Identify A and B given the Z scheme of the light reaction, students. You need to identify what is A and what is B. Okay. Now, students, first of all, tell me, students, in the chat right now, is this cyclic or non-cyclic? Is this cyclic or non-cyclic, students? You will tell me, sir, this right here is non-cyclic. In the case of non-cyclic, both the photosystems are involved here. Both the photosystems are involved here. Now, all of you should know, students, which photosystem is involved first, which is involved second here. Is PS1 coming first because it is first here? No. PS2 comes first, then PS1 comes. Okay. So what happens, students? First PS2 comes, A is going to be PS2, then we have PS1. So PS2 comes first and PS1 second comes, PS1 comes second. Next question to all of you, students. I want everyone to tell me in the chat right now. PS1 works at which wavelength here? P, which wavelength here? And PS2 works at which wavelength? P, 680 or 700? All of you in the chat. Which are the two wavelengths of your PS1 and PS2 in the chat also? Because in the examination, what they can give was, instead of writing PS2, PS1, they can give you the wavelength. So then the question becomes even more confusing because you need to know the wavelength also in such cases. You need to know the wavelength also in such cases. Okay. Now, students, next question on the board. Oh, very good question, students. Again, very, very good question on the board, students. Such questions you don't get easily. Very, very good question. Very, very good question on the board. Very good question on the board, students. You don't get such questions on the board nowadays, but very good question. Which is not true regarding the given diagram. Students, first of all, tell me, students, in the chat, what is the diagram here? What is this diagram? Is it the stem? Is it the root? Is it monocot, dicot, leaf? What is it? Students, first of all, tell me in the chat, what is this diagram first of all? I don't, first of all, understand diagram. Where is the even diagram coming from? Before answering the questions, all of you tell me in the chat, what is the diagram given here? What is this diagram? What is the diagram, all of you? What is this? What on earth is this diagram, all of you? Leaf? Are you sure, students? Is it a leaf? Stem? Root? Monocot? Dicot? What is it? Students, if you know the answer, if you know the sectioning, you will go crazy. This is a sectioning here. This right here is a TS of stem of monocot. This is a monocot stem right here. It is a monocot stem. It is not a leaf. It is not a root. It is a stem of monocot. Now you answer the question, students. A large number of scattered vascular bundles is correct here. Each vascular bundle, okay. Vascular bundles are conjoined and closed here because it's a monocot closed. Yes. Phloem parenchyma is absent here. Yes, phloem parenchyma is absent. But again, students, again, each vascular bundle is surrounded by collenchyma. So all of you should know, students, collenchyma is absent in the case of monocot. Collenchyma is completely absent in the case of monocot. Yes or no? Yes or no, students? So here, what happens here? If collenchyma is absent here, so what do we have? In the case of collenchyma, we have sclerenchyma. Sclerenchyma is present in the case of your monocot stem. Sclerenchyma is present here. Students, how was the question? Students, how was the question? How was the question? How was the question? Quickly in the chat, all of you. Good question or good question. This is the quality of questions you can expect in your NEET 2025. See, yes, there will be certain easy questions, but these are the questions which can decrease your marks, which can make sure your marks will go down. Now, students, when you are solving such questions, all of you will have some idea where you're going wrong. Tell me yes or no. All of you will have some idea where you're going wrong right now. So I want all of you today, after today's class, I want all of you to sit and reflect on the topics which you are not able to solve. Again, go back to those topics, revise one more time. Okay. Next question on the board, student. Next question on the board. Next question on the board. Based on the description, identify the cell organelle. They have given you some certain hints here. Certain hints are given here. Based on these hints, you're supposed to know which is the organ they're talking about. Okay. Student, yes, students, RCB yesterday won. RCB won yesterday. Comfortable win. RCB yesterday. Plastered? Is it plastered? Now, students, again, you should be able to tell me, students, again, ribosome, protein synthesis, that is going to be RER, lipid synthesis, SER. So, endoplasmic reticulum, students. Endoplasmic reticulum. Okay? Endoplasmic reticulum, students. Next question on the board. Here we go, students. Again, choose the incorrectly matched pair here. Okay, you're supposed to tell me which is incorrect here. Aleoplast, oil and fat, correct here. Amyloplast, carbohydrates and starch, yes. Aeroplast is proteins, yes. Then what is the answer, students? What is the answer? Incorrect here. None of them are incorrect. None of them are incorrect, students. None of them are incorrect. Okay. None of them are incorrect, students. Done? Done, students? Done? Yes? Done, students? That is going to be option number D here. Very easy question. Very easy question, students. Very, very easy question. Nice question again, students. Which of the following is a representation of the diagram of cholesterol? Which of the following is a diagrammatic representation of the cholesterol, students? In the chat. All of students. All of students. Which is a diagrammatic representation of cholesterol in the chat, all of you? Now again, such question can come in examination. You need to be very careful in answering. This is cholesterol is very easy. Cholesterol is very easy actually. That is going to be your here on the board. Cholesterol is going to be option number B here, cholesterol. Now if they change the question, let's say they change to uracil, uridine, or let's say, forget it, they'll ask the question, what is adenine here? Tell me, students, which of the following is a purine here? Purine. Which of the following examples here is a purine, that is adenine here, adenine. By chance they ask, because this diagrams are given here in NCERT, so they might ask you here. Which of the following is a purine here? Okay. Then the answer is going to be option number 1. Then the answer is going to be option number A here. Okay, in that case. Now cholesterol is option number B. Okay. Next question on the board, students. Which of the following statements accurately describe the stages in the sewage treatment plant? You're supposed to tell me which is the proper order. Okay. First, primary treatment involves physical removal. Okay. Okay, through filtration, sedimentation, tank 1 and tank 2. All solids that settle in the form of primary sludge and the supernatant forms the effluent. Okay, then comes second, that is sedimentation tank. The effluent from the primary settling tank is taken to secondary tank, secondary treatment. That is biological. It is in order itself. It is in order itself. It is in order right now. 1, 2, 3 is in order. Proper order is given. It's given a proper order. Okay. Next question on the board, students. The Mendelian disorder caused by substitution of students. This diagram you can tell me easily, students. This diagram you can tell me easily. If they're giving the diagram, very easy. It's an example of point mutation. Example of point mutation, students. Yes. It's going to be sickle cell anemia. Sickle cell anemia is the answer. Okay, students. Now, that was your tricky question, students. That is the end of tricky questions. See, if I bring you more questions right now, there will not be enough time for your zoology, chemistry, and physics. Okay. So these are tricky questions for today. Students, if you want more tricky questions, you should tell me in the chat. You should tell me in the comment section. Not here. You should tell me in the comment section, sir, we need more tricky question from your side. We'll bring it. We'll make it happen. Okay, students. So now we come to the third part of today's class. All ready, students? All ready? Now, students, we'll be starting with AR questions right now. We'll be starting with AR questions. Shall we start, students? Shall we start? Students, shall we start with the AR questions right now? Shall we start with AR questions right now, students? Shall we start? Now, I want all of you to rate your, how did you do the tricky questions? How did you do the tricky questions, students? Out of 5, I want you to rate your tricky questions. Out of 5, I want you to rate your, how many were you able to get correct? How many were you able to wrong? Based on 5, right? Based on rating of 5. Based on rating of 5 students, how was your tricky questions part? Again, some of the questions were difficult, some questions were very easy there also. Okay, 2, 3, 5 out of 5, all correct here. 4 out of 5, 4.5, 3. Okay. Now, students, there were not of many tricky questions there because I did not add any of the numericals there. See, if I add numericals, it becomes difficult again. Okay. So, if you want a part 2, I'll make a part 2 also where we'll have more numericals. So, students, if you want that part 2 to happen, I want everyone to make sure this video gets 1000 likes before the physics ends. So 1000 likes your target right now, 1k likes your target right now before the entire video ends today itself. Okay, so if you make that happen, we'll make a part 2. Okay, we'll make a part 2 in that case, students. Now there's a question on the board, students. I want to answer the question on the board. Now, students, let's start with AR questions right now. Okay, now we're starting with AR questions right now. Okay, now algae are thalide organisms of the kingdom Plantae. Their body is differentiated into root, stem, and leaf. What is the answer, students? What is the answer? What is the answer, students? Plant physiology, students. I want to take, students. I want to take. Okay, midnight. Let's see, students. Students, what is the answer? Reason is not correct because, students, it is not differentiated. Body is not differentiated into root, stem, and leaf. Okay. Next question on the board, students. Next question on the board. Endomembrane system includes ER, Golgi, lysosomes, and vacuoles. Their functions are coordinated because they're part of endomembrane system, students. Their functions are coordinated, students. What is the answer? What is the answer? What is the answer? What is the answer? Quickly, students. Quickly in the chat. Students, again, these are very, very important questions. All of you need to solve them very carefully. Easy. This is an easy question. Functional coordinated. Next question on the board. Zygotine is characterized by formation of synaptonemal complex. Okay. Synaptonemal complex helps in synapsis of non-homologous chromosomes. Okay. What is the answer? Okay. Again, I will say this is a little tricky question because your one word, if you don't read properly, you can go wrong. Okay. Yes. What is the answer? What is the answer? Option number A. Option number A, students. Is option number A right? Option number A, option number B, C, D. A is right. A looks good for me because that is synaptonemal complex, right? A is correct. Students, again I am telling you, one word can change the answer here. One word can change the answer. Just 3 letters, 3 letters. Answer is option number C. Why, students? Because this is the sus here. If this was not mentioned here, then the answer would be different here. Okay. So it says non-homologous chromosomes. Okay. So please be careful what they're asking you here. Next question on the board. Here we go. Chloroplast, chromoplast, and leucoplast are variable forms of plastids with different coloration. Such classification of plastids is based on a different structure. So what is the answer? A, B, C, D. Option number C again. Why? Why option number C, students? Because based on the, see, this entire classification, the entire classification of leuco, chromo, and chloro is based off the pigment present here. Pigment. So it is based on the pigment here, not based on the structure here, students, not based on structure. Next question on the board. In mustard flower are hypokynous in nature. In mustard flower, other floral whorls are present above the level of ovary. Again, read the question properly. Read the question again. These questions are very important. AR questions will take your time, but solve them properly. AR questions might take the time, but solve them properly, students. Lisa, Sweetie, Chinnu, Rakshita. Again, I can see so many new students again. Bharat, Rishika, Nityashri, Ravindrani. Students, again, why C again, students? Why option number C again, students? Can you even tell me why option number C again? Because, students, here, this entire thing is going to be wrong. Because this is an example of, students, what? Other floral whorls are present above the level of ovary? No. It is present below. Ovary is superior here. Ovary is supposed to be superior, students. Everything is supposed to be below the ovary here. Okay. Next question on the board, students. Next question on the board. See, again, if you write fast, you'll be wrong. In dicot stem, endodermis is referred to as starch sheath. Okay. The cells of endodermis are rich in starch grain. Students, this is your starch sheet is found in the case of your dicot stem or root. I'm just trying to be cautious here. Students. Cautious. Root or stem? Just to make you confused little. Students. Root or stem? Yes. Stem only, right? Or root? Stem or root? C or A? A or C? A or C, students. Stem only, right? Very good, students. Very good. So don't get confused. Be confident in what you know. Do not get confused just because I'm telling it root. Okay. So students, here, let's say in the case of stem, I have dicot stem have endodermis is made up of starch sheath. Okay. Now what about, students? What about in the case of dicot root? What is so special about endodermis? What is so special about dicot root? What is so special about endodermis? Do we also have something special here in the case of your dicot root? Yes, we have. There is something called a suberin deposition. Because there's something called a suberin deposition, we have Casparian strips there. We have Casparian strips in the case of your dicot root here. Casparian strips. Remember that much. Next question on the board, friends. Photorespiration is one of the wasteful process. In photorespiration, there is no synthesis of ATP and NADPH. Very easy question, friends. Very easy question. Very easy question, students. Suberin Casparian strips. Very good, students. Suberin deposition. Karuna, Chintu, Anupama. Yes. Usha is our regular students. Rishika. This is right. This is easy, student. This doesn't require much thinking. Next question on the board. Kinetin is a synthetic cytokinin. Kinetin was obtained from herring sperm DNA. Okay. It was obtained from herring sperm DNA. Kinetin is synthetic cytokinin. Quick. Now again, students, this option is very controversial. Okay. Now students, this is correct. Both are correct here. But students, the option is B here. Why? Because listen to me. Why it's B? Listen to me. If this statement was given here, kinetin does not occur naturally in plant. If this statement was given here, then option A would be correct. Option A would be correct here. But they're not telling you here. They are not telling you, students. They are not telling you here that kinetin is obtained from, yes, it is obtained from herring sperm DNA, autoclaved and everything. But that does not tell you why that does not tell you it is synthetic in nature. If you want to tell it is synthetic in nature, then this is the reason here. This is the reason, student. This is the reason. Kinetin does not occur naturally in plants. That is why it is called as what? That is why it is synthetic in nature. Okay, so this is the reason here. This is the reason here, students. So what is the answer? The answer is option number B then. Okay, so there are certain lines of NCERT which they can play with you. Shobhi, yes, you can watch. Yes, you can watch. Okay, so option number B is the right answer. Option number B is the right answer then in such case. Okay, next question on the board. Next question on the board. Next question on the board. In majority of your angiosperms, a typical anther is, oh, this question, this is ma'am's question, student, by mistake it has come. Okay, in a majority of your angiosperms, a typical anther is bilobed. Each lobe has 2 theca. Each lobe has 2 theca, students. Each lobe has 2 theca. Again, very, very good question. It's a quality question. If you go for option number A, you'll be losing marks. Very, very good question again on the board. Students, again, both are correct here. Both are correct here. But this is explaining dithecus here. Here we need to understand why it's called bilobed. It is not explaining bilobed condition here. It is explaining dithecus condition here. So option number B is right answer. Yeah, option number B is right answer. Ma'am's question came by mistake, students. Okay. Now, students, again, next question again. This is a question which is a direct line of NCERT and many of you might go wrong here. In terrestrial ecosystem, a much larger fraction of energy flows through detritus food chain. Reason? Because in aquatic food chain, GFC is a major current of energy. Again, you might even not know if the assertion reason both are correct also here. That also might become confusing here, students. That also might become confusing here. That also might become confusing here, students. What is the answer? What is the answer, students? Yes, you can do the 3 hours marathon. Yes, you can. Students, students, tell me first of all in the chat, is this even correct here? Is this even correct? Is the reason even correct here? Students, is the even correct here? Tetrathecus is 4 theca. Yes, not dithecus. Dithecus is going to be 2 theca. See, this is bilobed condition. In one lobe there are 2 theca. Bilobed condition, 2 lobes. Every lobe has 2 thecas here, right? Friends, let me know the answer in the chat. Dun-dun-dun. Students, all of you should know this very well. All of you should know this very well in the chat right now, students. Never forget this. Okay, I hope it is visible to all of you. In aquatic ecosystem, GFC is the major conduct of energy. So in aquatic system, obviously, in aquatic ecosystem, we'll only have GFC mainly. Because in aquatic ecosystem, detritus food chain is not there much because aquatic, right? Detritus is very rare because only the bottom side might be there. But majority is water, right? So detritus is not going to be there major. Okay. Now, students, in terrestrial ecosystem, much larger fraction of energy flows through the DFC. Okay, flows through DFC. So, in the case of terrestrial, DFC is more. In the case of terrestrial, DFC is more always, students. DFC is more. Okay. So, answer is both are correct here, but it is not explaining here. Option number B is the right answer. Both are going to be correct here because in aquatic GFC is, yes, GFC is more. In terrestrial, DFC is more. Okay, DFC is more. Detritus food chain is more in the case of terrestrial because terrestrial we have more land. We have more land, so more detritus food chain. Okay, next question on the board, students. Competition occurs between closely related species competing for the same resource that are limiting. Gauss exclusion principle almost starts from here only. Okay, totally unrelated species could not compete for the same resource. So do you think unlimited species also compete for the same resource? Unrelated species also compete for the same resource? Yes or no? Or is this wrong here, students? Or is this correct here? What is the answer? Students, what is the answer? Or is it option number B? Option number B or C? Option number B or C? Option number B or C? Tell me, students. Option number B or C? Quickly. Or D? Students, option number, why is it given? Students, option number C is the right answer. Students, wrong, it's given here. This is, okay, this is correct here. Okay, A is correct, R is wrong, not D. A is correct, R is wrong. Okay, because students, totally unrelated species also can compete for the same resource. For example, can anyone give me an example? Can anyone give me this? Can anyone give me this example here? Why see this example for this example for students? Example, flamingo and the fishes. Flamingo and the fishes fight for the same zooplanktons. Flamingo and the fish fight for the same zooplankton. Flamingo and fish are unrelated. Flamingo and the fish are unrelated, students, unrelated. Okay, so unrelated species can also compete. Unrelated species can also compete. So C is the answer. Okay. They compete. Unrelated species also compete for the same resource. Next question on the board, students. Yes, flamingo and the fishes. Next question on the board. Snake and cat for the right. Exactly, sweet. Exactly. Snake and cat are not related, but they same fight for the same. They fight for the same American lake. Exactly. Yeast as Saccharomyces cerevisiae are used in baking industry. Carbon dioxide produced during fermentation causes the bread dough to raise by thermal expansion. Easy, students. Easy question. Easy question. Next question, students. In bacteria, mesosomes are formed by extension of plasma membrane. The mesosomes in bacteria help in DNA replication, cell wall formation. Cell wall formation, students. What? Someone is sending super chat. I'm seeing this for the first time. One second, students. 1 second. Let's go back and clear this confusion. Actually, students, let me tell you one more thing here. Okay. Actually, according to NCERT line, students, resource, this actually is limiting also is wrong actually, because sometimes what happens, if the resource is not limiting also, there will be still competition. Not limiting can also be, not limiting can also be, there will be still fight. Not limiting can also be fighting also. Okay. Not limiting can also be correct here. So this also becomes false actually. Yes or no, students? Yes or no? Because I've told you always, even if the resources are not limiting also, they can still be fight also. Okay. So let's just introspect on the questions. Students, actually this has to be B actually, I feel. Because is it explaining why it is used in thermal industry? Yeast such as Saccharomyces cerevisiae are used in baking industry because carbon dioxide, no, I think this has to be B, student. This has to be B. This has to be B, not A. This has to be B because it does not explain why it is used in baking. No, it has to be A, student. 1 second, student. 1 second. Baking, right? Baking industry. This is A. This is A, student. This is A. This is A itself. This is A itself because dough bread baking industry. I read it as brewing industry. Students, if they make it as brewing industry, then it is B. Then it is B. Since it is saying baking. Students, one super chat and the entire thing became lagging. Student, is it proper right now? Is it proper right now? I am here only. Okay, perfect. Next question on the board. It's fine, right? Students, see that one super chat and the entire thing became entire. Student, next question on the board. The appearance of recombination nodules on homologous chromosomes during meiosis occurs, students. recombination nodules, or it could be happens in packaging. It happens in packaging, students, but at the site of crossing over, at the site of crossing over at packaging, site of crossing over packaging, students. Okay. No. Next question on the board. Here we go, students. Here we go. In the enzyme, which catalyzes the breakdown of your entire reaction? They're asking you which is the porosity group here. They're asking you, students, which is the porosity group in your hydrogen peroxide reaction? Which of the following is the porosity group here? Okay. Which of the following is the porosity group in the case of your hydrogen peroxide reaction? The porosity group. Porosity group here. Heme is right, students. Heme is right answer. Heme is the right answer, students. Now, again, students, in your entire these two concepts are very important. Biomolecules, all of you should remember. All of you should remember these two tables, because multiple times question have come from these two tables. These two tables becomes very, very, very important. Okay, very important tables. Now, students, next question on the board. Here we go. Students, I have 2 more questions and we are done. I have 3 more questions and we are done, students. I have 3 more questions and we are done after that. Okay. How many ATP and NADPH are produced, are required for synthesis of 1 molecule of glucose during Calvin cycle? Of glucose during Calvin cycle, students, for glucose. How many molecules of ATP and NADPH are required? Okay. This all of you are by-hearted by now. This all of you are by-hearted right now. I will change the question. I'll change the question to wait. I'll change the question. Okay, I'll change the question to some other question, students. How many CO2 molecules are released per glucose in Krebs cycle? What is the answer? What is the answer? How many molecules of CO2 are released in Krebs cycle? Only in Krebs cycle. Do not take no link reaction, nothing. No link reaction, nothing. Only in the case of Krebs cycle, how many molecules of CO2 are released? Okay. Sir, your writing is good. From which angle this is good, students? Students, all the students who are telling the answer is 2 are gonna be completely wrong. 2 is wrong here. 2 is wrong. Why? Students, the question says what? The question is telling you in one Krebs cycle, right? For one acetyl CoA, there are 2 CO2 released. This acetyl CoA, one acetyl CoA is coming from one pyruvate. But when I told the question per glucose, one glucose will give you 2 pyruvate. So one glucose will give you what? 2 acetyl CoA. And one acetyl CoA is giving you 2 CO2. So 2 acetyl CoA will give you 4 CO2 is the answer, students. 4 CO2 is the answer. So what is the answer, students? What is the answer? Students, what is the answer? Okay. Students, what is the answer? So answer is going to be 4. So all the students. So what is the answer, students? The answer is going to be 4. Clear? Students, if you want more such questions, tell me in the chat later on. Here is the question on the go. Here we go, students. Identify the correct set of statements regarding with the properties of humus, not hummus, humus. Okay. What batch? Hmm. This is going to be option number A is right answer. Highly resistant to microbial action, dark color, no. Yes, no. Students, last question from my side. Here we go. Let's see who can tell the answer, students. Last question from my side. Okay. I am surprised, obviously. First time ever super chat. I don't even know super chat. I didn't even know we had enabled super chat. I don't think so it was enabled also. From nowhere it came. No, students. Question on the board. Here we go, students. Question on the board. Students, I should ask you one more question like that. Can anyone tell me how many ATP are produced in fermentation process? In the chat. How many ATPs are produced in fermentation process, students? Yes. What is the answer? How many ATPs are produced in fermentation? Students, I'll give you two more questions and I'll go. Gopika Mam is waiting there on the other end. Two ATPs. Two ATPs are produced. Okay. Now, students. Glucose, that is there. 1 CO2, that is there. Students, I will make a list. I'll make such questions and come next time, students. I'll make a list of questions and I'll come next time, students. I'll make you some difficult questions and come next time. So until then, students, keep solving questions. I hope it was useful to all of you, students. I hope it was useful to all of you. My last question to all of you is, can anyone tell me, students, which is the final electron acceptor in the case of alcoholic fermentation? That is my last question for all of you, students. That is my last question for all of you. Which is the final electron acceptor in the case of alcoholic fermentation? Last question from my side. Last question from my student. Last question from my side. Which is the final electron acceptor in the case of alcoholic fermentation? Okay. Students, thank you so much. Bye bye, students. Take care. I'll just call Gopika Mam. Gopika Mam will come right now and we can go. Okay, students. So I hope all of you enjoyed. All of you students, if you're joining late, here is a small review what we did today, students. Here we did it in 3 parts: easy, direct questions, tricky questions, AR questions. All of these questions will be available on Telegram channel. All of you on Telegram, right? If you all the questions will be available on Vedantu NEET English Telegram channel and also on Baswaraj Sir Biology. Also on my Telegram also, everything will be available, students. Also on my Telegram channel. Now today, students, if you go to my Telegram, all the previous notes will be uploaded today itself. So if you go to my Telegram channel, students, every single notes will be uploaded today. Plant physiology, all 3 plant physiology, all 11 PYQ, 12 PYQ, then entire class 11th botany, entire class 12th botany, every single notes will be available on my Telegram today itself. So go on, download all the notes, clear all the doubts and keep studying, students. Keep the grind on. Okay. So let me just call Gopika Mam, students. Let me just call Gopika Mam.
Hello students, welcome to Vedantu NEET English. I'm Miss Gopika, your own biology master teacher. I hope I'm audible and visible to all of you. So all of you did a wonderful session on botany, right? You did easy question, tricky question, AR questions. It's a mix of everything, right? So now for zoology, we are going to do some easy questions, medium questions, difficult questions. So it's going to be a mix of everything, okay? Yes, no break. No break. Why do you need break after 1 hour of paper? You're going to write a 3 hours paper. You do not have the, you cannot even think of asking a break right now, right? After 3 hours, if you ask a break, yes, it makes sense. After 1 hour break, huh? It's time to break all our bones, right? Hello, good morning, good morning. How are you all doing? How are you all doing? Yes, I think all of you are doing great. I saw all of your energy. I want more energy in zoology because questions are really easy. Okay, it's not that hard because I did a revision for you very recently about human physiology, genetics, everything, and I have been doing continuous classes of question practice as well. If you go back and see, all my question practice videos are there, top 50 questions, NCRT, decode, or even Tejas series. So, lot of question practice we have done in zoology. So, all you have to do is answer the questions confidently and make me proud. Okay. Ready, all of you? I hope all of you marked your attendance. Okay, energy is going to be high throughout. Full time chat should be moving. Okay, yes. Okay, chalo, let's start. Good morning, good morning. So first question on the board. Okay, not on the stage, board. Yes, first question. Which of the following is odd one? Okay, odd one with respect to organ system level of body organization. Platyhelminthes, arthropods, annelids, mollusca. Which of the following is the odd one out in basis of what? Organ system level of organization. So cellular level is there, tissue is there, organ level is there, organ system level is there, right? Yes, come on, all of you quickly answer. I think there can be a light difficulty with the live. Okay, but don't worry, it is perfectly visible to all of you. Yes, perfect. Okay, quickly smash the like button as well. Let's make it 500 likes soon. Okay, yes, excellent, excellent. The answer is going to be none other than platyhelminthes, because platyhelminthes is the only one who is falling under organ level of organization, and the only fellow who is falling under cellular level of organization. Can anyone answer cellular level of organization quickly in the chat box? Come on, come on. Quickly answer. Perfectly fine. Yes, thank you. Yes, 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 yes. Okay, some of them would have left thinking that, oh my god, live got ended, but students, don't worry, live is still there. Okay, yes, let me just inform them as well. Please answer this question by now. Faster, 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 faster. Very good, very good. Yes, excellent, excellent. Porifera, right? Yes, good job, good job. Yes, okay. Fine, perfect. Next question on the screen. Okay, incomplete cartilaginous ring are present in tracheae, secondary bronchi, tertiary bronchi, respiratory bronchi. Okay, what are they talking about? Incomplete cartilaginous ring. That means on the dorsal side, the ring is going to be incomplete, so we can prevent the collapsing of the muscles, right? So this way, which and all parts have that. Trachea have secondary bronchi, tertiary bronchi, respiratory bronchioles. Come on, quickly in the chat box. All of you. Answer. Answer, answer, answer. A, 1, 2, 3, 4. Tell everything. Tell everything. Okay, very good. Very good. What is going to be the answer? Ma'am, trachea is the one that we studied always about having incomplete C-shaped cartilaginous ring. Secondary bronchi also will have, tertiary bronchi will have, bronchioles are not going to have. So the answer is going to be option number 2. Excellent, students. Do you want to keep a track of marks? We can do that. Do you want to keep a track of marks? Come on, +2 in the chats. I think if you keep a track of marks, because a lot of questions are there, it would be easy for you, and also you will have the motivation. Yes, I got it. Okay, +2 pakka. Okay, yes, let's go. Okay, next question. Easy questions, okay, don't worry. True coelom is lined by ectoderm, mesoderm, endoderm, ectoderm and endoderm. Okay, what is lined by? True coelom is lined by ectoderm, mesoderm, endoderm, ectoderm and endoderm. Come on, quickly in the chat box. All the questions are going to be high weightage. Animal kingdom is a high weightage chapter for all of you. Please understand, 20 marks comes from that chapter. Okay, so there's no way that you're skipping animal kingdom and thinking that, okay, one question might come. No, no, no. 5 questions can come. B for biology by Gopika, pakka. Okay, those who want to join my Telegram channel, you're always welcome to join my Telegram channel. The name is Gopika Biology Official. Okay, we have changed this name to Gopika Biology Official because many students are finding it difficult. Okay, so all the notes will be available here. All the zoology notes are already on the channel. I mean, and if you want any notes specifically, please let me know in the comments. I will share it with you. Okay, all my handwritten notes. You know, no, I write handwritten notes, so all those notes are available. Yes, excellent students. Mesoderm. Okay, now what happens in the case of what happens in the case of pseudocoelom, students? Pseudocoelom, what is going to happen? The mesoderm is going to be lined, but it is going to be in the form of, yes, Gopika Biology official. Yes, very good. Very good, all of you. Okay, tell me the answer this. Pseudocoelom. Pseudocoelom means what? False coelom. So if false coelom, ma'am, it is lined by mesoderm, but there's a problem. What is this problem? The problem is that, yes, excellent. It is in the form of scattered pouches. Okay, scattered pouches. Okay, so I'm going to give you this also 1 mark. So +4 in the comments. Come on, +4. Excellent. Ascalminthus have pseudocoelom. That is why we remember the trick VA, because they are excellent. They're different from everyone, right? VA. Okay, excellent students. Moving forward to the next question. In most simple type of canal system of Porifers, which of the following shows the correct water flow? Okay, correct water flow you have to tell me in the most simple type of canal system. That means Porifers. Okay, so option 1, option 2, option 3 and option 4. Now all of you look into the option and please let me know the answer. Yes, wow, very good. Wow. Yes, tell me quickly in the chats. Canal system of Porifers. Okay, your answers might come a little late, but you people know the answer. Yes, excellent students. The answer is going to be ostia, which is a small opening, right? Through this the water will enter, there will be a cavity and then there will be a big opening again through which the water will exit out. Okay, students, what is the one thing that spongiocele has? What is that one specialized cell or component that spongiocele has? Come on, quickly in the chats, that also you can answer and you can take your 6 marks. Okay, you can take your 6 marks if you answer this. If you don't answer, 5 marks. Many people are telling A, no, it feels like you're singing. Okay, so that long A you people are sending. Huh? Yes, excellent. No. You're telling me about the skeleton. That's not what I'm asking. I'm asking about choanocytes. Very good. Some of you got it right. So, choanocytes or collar cells, right? This is what I'm asking all of you. Collar cells, okay? Yes, choanocytes or collar cells. That is what I'm asking. Some of you told me spongion fibers. That is the skeleton. Specules and spongion fibers are the skeleton. So, how many of you got +6? Quickly let me know in the chats. Moving on to the next question. Yes, very good. Okay. Now, column 1 and column 2, we have jawless vertebrates, placoid scales, ctenoid scales, cloaca, mammals, ostrictus, amphibians, cyclostomes and chondrichthyus. Come on students, do the match the following. Do match the following, all of you in the chat box. Yes, collar or choanocytes. Yes, very good. Collar cells or choanocytes. Okay. Yes, very good. 1, is it? Okay, let's try. What's the answer? Jawless vertebrates are who, students? Obviously, it is going to be cyclostomes. Tell me that you solved only with that. you people are supposed to go further, okay? Such lazy people. Placoid scale, obviously cartilaginous fishes. Ctenoid scales can be present in bony fishes. Cloaca are going to be a characteristics of amphibians, okay? So, it is going to be, I know with one option you people did, okay? All shortcuts. All shortcuts in life. I know. That's why so fast answer came, okay? +7. Quickly in the chats. Next question for all of you. Many of you go wrong in this question, so please try to answer correctly. Bidder's canal in frog would be present in the cross section of. Okay? If I want to see Bidder's canal, if I take a cross section of what? Testis, kidney, ovary and brain. Where will I see the Bidder's canal? Come on, answer me. Quickly. D option, A option, I knew it. That's why answers came so fast. This one is 2, is it? Yes, very good. It is kidney. Many students get confused with testis. Why? Because in reproduction topic only we talk about Bidder's canal. How it is helping in the transport of the sperm. But please do understand, Bidder's canal is a part of the kidney, not a part of the testis. Okay? Excellent students. I'm so proud of all of you. So +8 in your pocket. Okay? +8 in your pocket. Next question, it's a diagram based question. Examine the figures of diploblastic and triploblastic organizations in animals given below and identify the labeled parts A to D. Okay, A to D you have to label. I hope it's a very clear diagram. The A, B, D and C. Come on, quickly in the chats, answer. Yes, very good. Most of you got +8. Excellent. I told you questions are going to be easy. There's nothing to be worried about. This one, can you answer? Let's see. A, okay. B, okay, perfect. C, yes. D, yes, I got it. Did you get? Ha, very good, very good. It's going to be option number, D. Why? Because this outer layer is going to be ecto. So students, anything outside is always going to be ecto, right? And B is going to be the innermost layer, which is endo. C is going to be what? An undifferentiated layer, which is called mesoglia. And last option we have D, which is in triploblastic, that is mesoderm. Okay, mesoderm, right? Excellent. So how much marks right now? +9. +9. Okay, +9. Good job. Moving on to the next one. Match the following column 1 and column 2. Okay, easy question this is. During contraction of muscles, during relaxation of muscles, thick filament, thin filament. Actin, Z line moves to the original position, Z line pulled inwards, myosin. Very easy question. Yes, mesoglia, very good. Okay. Oh, one option. All shortcuts. All shortcuts my kids have learned day by day. Okay. People are becoming. That's all, huh? That's your NEET score, huh? 454 is your NEET score. Huh? That much only you want in your NEET, is it? That's why people are not liking the session. At least 720 should happen, no? Come on. All of you. All the conjuice. Yes, A1. Okay, very good. During contraction of muscles, the Z line is going to move inwards. Students, same thing. One more question can come. They can ask you about the A band. They can ask you about the I band. Okay. They can ask you about ozone. Don't worry, you are going to practice all kinds of questions because we are doing an entire question series. So there are going to be tricky questions. There are going to be AIMS JIPMA level questions. There are going to be, tomorrow we are going to do one of the most interesting one where maximum students make silly mistakes. So tomorrow's session, don't miss at any cost. Okay, tomorrow we are going to select an entire set of questions where we saw plenty of students over the year making the same mistake. Okay, because how do you know this, ma'am? Because in our batches we have students, they write mock paper and we have noticed that these questions, most of the students make mistake. So do not miss it by any chance. Okay, yes. No, same questions will never come for any examination. No competitive exam questions are going to be repeated. Only the pattern might be the same. Okay, pattern, concepts are going to be the same. I think by now you should know this much. Famous creator. You're not famous enough with the knowledge, I guess. It's okay. A band, I band, ozone. Okay, all this they can ask you. Are they constant and all of that? So very good, all of you. So right now we have reached +10. Okay, +10. Very good. Excellent. Okay. Aha, controversy and all you people are telling. Yes, very good. A will remain constant. Okay, A will remain constant, Sneha, because I'm asking middle other questions as well. That is why you got +10. Okay, she has a problem that the question marks are more. First time I'm seeing. Okay, head zone, what will happen? Head zone will get reduced, ozone will increase. Okay, head zone will get reduced, ozone will increase. Very good. Very good. Okay, perfect. Moving on to the next question. Protonephridia or flame cells, or this is not there for you. Okay, are the excretory structures in? Platyhelminthes, flatworm, rotifers in some annelids, cephalochordates, and all of the above. All of the above. Come on, quickly answer. Okay, protonephridia or flame cells, or are the excretory structures? Okay. Come on. All of the above. Oh. No. Yes, very good. All of the above is wrong. Students, here all of the above question is given as a wrong statement. Okay. Your answer is going to be only platyhelminthes, because here we are talking about protonephridia and flame cells. Flame cells are a part of who? Your flatworms or platyhelminths, right? So the answer is going to be option number A. So that gives you 11 marks. Okay. Students, what about annelids? Tell me, what about annelids? What is the excretory organ in annelids? Quickly in the chats. Excretory organ in annelids. All of you will be able to answer this easily. Yes. Very good. Not 2. It's option number 1. Option number 1. Come on, come on, come on, come on, come on. Faster, faster. Yes. What is the excretory organ in annelids? Annelids, what the excretory organ? Excellent. The answer is going to be nephridia. Yes or no? Nephridia is the answer. Yes, very good. Excellent. So that gives you plus 12. Okay, any question you're answering, you're going to get marks. Okay, what will I do with your marks, students? I want to give you all the marks. Goblet cells are unicellular, bicellular, multicellular, acellular. Unicellular, bicellular, multicellular and acellular. Come on, quickly answer this in the chats. From where is this question coming? This question is coming from animal tissues. Okay, animal tissues like this. Okay, animal tissues question it is. So come on, quickly answer this as well. Excellent, excellent. Goblet cells are unicellular, right? Goblet cells are unicellular. Now tell me this. Goblet cells are a modification of which cell, students? What kind of an epithelium are they? You can say what kind of an epithelium are they? Or you can say which cells modify to form this kind of an epithelium. Any one you can answer. Or both also you can answer. Come on, all of you. Digestion is removed. Neelam. Come on, come on, come on. No, 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 no. What are you telling? Transitional? Student, that is compound epithelium. Compound epithelium, you have never studied any modifications. Yes, excellent. So, some of you are telling me which kind of epithelium? Glandular epithelium. Okay, glandular epithelium. Excellent. Some of you are telling me, ma'am, it can be a modification of your columnar and cuboidal. Okay, cuboidal. Yes. So here again, depending on the location, these people are going to be different. If it is like stomach, intestine lining, columnar is going to be there. Okay. So columnar and cuboidal and glandular. Yes. Excellent. Excellent. Okay. Perfect. Perfect. Digestion is not there. No, I thought you were telling me, ma'am, where is digestion chapter? So plus 13, 14. Okay. Whoever answered one among this will get 1 mark. Okay, some of you have answered some different, okay, different things. Okay, those who do not know, try to learn it now. Goblet cells are an example of unicellular glandular epithelium. Glandular epithelium has 2 major classifications. One based on the numbers, that is unicellular and multicellular. Second one based on their secretion, exocrine and endocrine. So please do not make a mistake, okay? Please do not make a mistake. Goblet cells are unicellular, salivary glands are multicellular. They are a modifications of your columnar and cuboidal epithelium depending on where their locations are, okay? Perfect, all of you? Yes, okay, PYQ session, good job. Okay, next question on the screen for all of you. Epithelium found in the walls of blood vessels and air sacs is columnar, squamous, cuboidal, ciliated. Epithelium found in the walls of blood vessel and air sac. Students, let me tell you, in animal tissues, all your questions are going to be based on functions and locations. Okay, so don't waste your time in trying to mug up all of this. What you can maximum do is know the locations. Clear? Come on. Excellent, excellent. It is going to be squamous epithelium because that is where your blood vessels. Anyone has a confusion? Air sacs. Remember diffusion. In diffusion, students, please tell me what are the respiratory layers? Okay. What is the respiratory membrane or the respiratory or the diffusion membrane? Come on, quickly in the chats. It's made up of 3 layers. What are those 3 layers? I hope all of you are understanding what I'm asking. This is what I'm asking. Okay, alveoli is there, blood vessel is there. I want the 3 layers. Quickly in the chats. Quickly in the chats. Yes, just give me the first 2 words also. It's fine. Like you can write, I will not tell you. Like you can write short forms. You know, no, you don't have to write the whole thing. You can just give me the short forms quickly because that question also can come for all of you. So I would request all of you to know, remember that every time when you're studying something about alveoli, I want you to remember the respiratory membranes. At the same time, in ultrafiltration, urine formation also, you have to remember the 3 layers. Excellent, students. Some of you are confused, I feel. Yeah, it is going to be squamous epithelium, basement membrane, endothelium. Now, when we go to ultrafiltration, what is going to be the case? Endothelium, basement membrane, epithelium. That epithelium is going to be the podocytes, which are arranged in intricate manners, which have filtration slits. Understood? So, please remember diffusion membrane and ultrafiltration membrane separately. Don't get confused. So, here you will see the epithelium, here you will see the endothelium, and here you will see the non-cellular layer, which is BM, basement membrane. Okay, so how many marks right now? How many marks right now? Quickly tell me in the chats. Wait. Okay, 14. Here 15, 16. Okay, 1 mark for this. 1 mark for this. 16 marks. 16 marks. Come on. Yes. Moving on to the next question. Very good. Okay, Rani has kept a track. Mucus, saliva, earwax, oil, milk and digestive enzymes are secreted by exocrine gland, endocrine gland, heterocrine gland, compound gland. Come on, easy question again. Okay. Yes. Mucus, saliva, earwax, oil, milk and digestive enzymes are secreted by exo, endo, hetero, compound. Yes. Very good, students. Very good, students. It is going to be exocrine glands, right? That means the glands which have ducts. Okay, the glands which have ducts. Endocrine glands means the glands that do not have a duct. They are ductless glands. They will add their secretions directly into the blood. Clear? Excellent. Okay, excellent. 17. Moving forward, which of the following, okay, 1 second, yeah. Which of the following is involved in rapid transfer of ions, small molecules and some big molecules? Tight junction, adhering junctions, desmosomes, gap junctions. Okay, come on. Yes, ductless glands. Very good. Very good. Which of the following is involved in rapid transfer of ions, small molecules and some big molecules? Tight, okay, adhering junctions, desmosomes, gap junctions. All these are what? Cell junctions. Everyone has one job. In that you have to study about 3 of them. So your answer is going to be excellent. Your answer is going to be gap junctions, because they are going to help in rapid transfers. Tight junctions are going to prevent leakage. Adhering junctions are going to do the cementing. Okay, desmosomes you don't have, but again they also help. They are also part of the cell junctions. Okay, they are going to help in transfers at the same time and also connections. Very good, students. Plus 18. Plus 18. Moving forward to the next question. Triangular head of cockroach is formed by the fusion of. Triangular head of who? Cockroach. Okay, it is formed by the fusion of. Come on, quickly tell me in the chats. Quickly tell me in the chats. Okay, I have a throat pain. That's why my voice is like that. Okay, it's not because I'm not drinking water. Yes. Excellent, excellent. Students, cockroach head is located at dash angle to the body. Dash angle to the body axis. Come on, quickly tell me in the chats. Students, we are going to do all the in-text questions, bold questions. Okay, bold, in-text, dates, scientist, all this very soon in this week itself. Okay, this week itself we are going to do it. So don't worry. I know many of you are thinking, ma'am, can you please do the bold questions? Yes, we will definitely do all the bold. Okay, in-text, scientist, dates for all of you, just like last year. So do not fail to join the live. Okay, yes. What a ASMR, that loud it was. Yes, answer this. Very good, Shrishti. Yes. Okay, Rani also very good. Yes, very good, very good. And Partha, yes, it is at right angle. Right angle, or we can say, because we are all intelligent in maths, we can also say 90 degree. Okay, because we are the head of maths department. Yes, very good. So 20 marks in your pocket. How many of you got 20? I'm excited to know how many of you got 20. Come on, quickly in the chats. And moving on to the next question. Abdomen of cockroach consists of how many segments? Okay, how many segments? Abdomen of cockroach consists of how many segments? 10, 5, 50, 100. 100. Let's go for 100. Come on. Yes, students, tell me. Okay, abdomen of cockroach consists of how many segments? How many segments? 20. Wow. Wow. 10 segments. Okay, wow. 1 minute. Yes, very good. Very good. It has 10 segments. Okay, now I told all of you that students, when you are preparing for cockroach, I want you to make a separate sheet. Okay, just make a paper like this and write all the numbers that are mentioned. Ma'am, what do you mean exactly by numbers? See, they're talking about head, okay, 6 segments. They're talking about abdomen, 10 segments. They're telling about location of anal cirri, okay. They're telling about how many out of the 13 chambered heart, how many of them are scattered to abdomen and thorax, okay. They're telling about the they're also telling about anal cirri, we already told. Yeah, they're also telling about the mushroom gland, the testis, the ovary, the collateral gland, okay, the spermatophore. So I want all of you to take a sheet of paper, write all the numbers mentioned in cockroach, okay, whichever segments they are talking about, boat shape segment, brood pouch, like that. N number of numbers are there. And what is going to happen when you're going to the examination hall, when you're studying so many topics, students, there are chances that you forget the numbers or mix up the numbers, okay. So I want someone to do that, okay. Yes, ganglia also they're asking, okay, how many categorizations are there. So please take a sheet of paper, write it, and if possible share it on my Instagram channel so I can share it on my Telegram channel, which can help other students as well, okay. If any of you are writing it very nicely with different types of colors, please share it on my Telegram channel. I will definitely post it on my Telegram, okay. On my Instagram you can share, I'll post it on my Telegram channel. Clear? Yes. Gopika Biology Official is my Telegram channel name. You can just search Gopika and you will be able to see it has around 6,700 students right now. Let's make it 10,000 very soon. Okay, with all your support, I can definitely make it 10,000. Okay, I started it very small, all of you know. Okay, my old students know it. Very small we started, so now we can go up to that much. Yes, come on students. Next question on the screen. See, again a number is coming. In female cockroach, which segment of the sternum is boat shaped? Again a number. Number based questions might look easier now, but in the examination hall it can definitely lead to confusions. Okay, yes, written. Okay, Meghna, you can definitely share with me if you're on Instagram. I think you're on Instagram, right? Yeah. Only frog in the syllabus? No dear, they have given still cockroach also, like they have given insect in bracket. So we are all going to prepare you for insect. The insect is going to be cockroach, okay? And cockroach is very easy, so there's nothing to break your head for. Yes, 17th segment, that is going to give you 21 marks, right? Students, 21, right? Did I miss any marks? Yes, I did miss 1 mark. So 22 marks. Okay, 22. Then later one student will be there in the comments. My God, ma'am, it is 22, ma'am. You put 21. I lost 1 mark because of you. Yes, 22 marks. Excellent. Moving on to the next question. Oh, 3 questions are there. Okay, 4 questions are there. So let's answer one by one. Okay, let's answer one by one. Come on. The alimentary canal of cockroach present in the body cavity is divided into 3 regions. They're talking about what? They're asking you about the alimentary canal. Okay. So here you will get 1 mark. So that will be 23. Okay. 24, 25, 26. These are lines of NCRT. So I've made fill in the blanks out of lines of NCRT. Best way to revise. Okay. Yes, we have got a target. Come on, answer. Yes. Quickly, come on students, quickly smash the like button. Why are we not yet near to the target? Okay, so much conjuicy shouldn't be there. Come on, 10 more to reach the target. Foregut, midgut and hindgut, excellent, excellent. Okay, good job. Foregut, midgut and hindgut. Next question. The mouth of cockroach opens into a short tubular pharynx leading to a wide tubular passage called as esophagus. True or false? True or false? So they are telling the mouth of the cockroach is opening into a pharynx leading to a wide tubular passage called as esophagus. So all of you remember crop, gizzard, before that esophagus was there. Was there a pharynx? Try to think and answer. Try to think and answer. All mixed answers are coming. Yes, true. Very good. Okay, true. Yes, they do have a short tubular pharynx and it opens into the esophagus. Now, hepatic CK is also known as what? Come on. Yes, excellent. It is also known as gastric CK. It is also known as gastric CK. Can you tell me the number also? Number of tubules so that you can get this 1 marks. Okay. Number of tubules, students. Anyone in the chats? Huh? Yes. Students, this wide word is there. No, it should be narrow, right? Narrow tubular. We have to check the lines of NCERT. Has they mentioned narrow? Just check NCERT and let me know. If they have mentioned narrow, then this question is going to be a false question. Okay? I want all of you to check and let me know if the word narrow is mentioned. I'm not able to remember exactly. Okay? If it is narrow, then it is false. Okay? Yes, this is going to be 6 to 8 tubules. Excellent. A blind 6 to 10 blind tubules called hepatic CK is present at the junction of the foregut and midgut which secrete digestive juices. Now you already know the answer of this. Narrow is mentioned. Yes, Pakka. Narrow means then this is false. Okay, I'm not able to remember exact lines of NCERT. So this is going to be false. Okay, because here the word wide is there. Okay, so when we read the question, we have to be more careful. So that is going to be false. This is also going to be false. So most of you made mistake in this. Okay, so you are going to get how much? 23, 24, 25 and 26 itself. People who made that also correct will get 27. Okay, so some of you are going to get 26, some of you are going to get 27. Good job, students, good job. Yes, moving on to the next question. The pituitary gland is located in a bony cavity called dash and it is attached to the dash by a stalk. Very nice question. Very nice question. All of you answer. I've taught you this in chemical coordination, at the same time in locomotion and movement. Okay, locomotion and movement. Yes, be honest with yourself. Very good, Madhumati. 26 marks, right? Yes, 26 marks. Good job. Okay, those who made mistake, please cut 1 mark from yourself. Be lenient, okay? Yes, very good. Wrong. Hey, what are you people telling? Dorsal side of the forebrainer. Yes, very good. The answer is going to be sella turcica. Sella turcica is present on which bone, students? If you answer this only, you're going to get full marks. Or else I'm going to cut your marks. So 27, 28, okay? You will not get 28 if you don't answer me. Where is sella turcica present? On which bone is it present? You have studied in locomotion and movement. Come on. Let me see. Oh no. He told, huh? Okay, I have to be careful. Thank you, Christina, for inside informations. Okay, very secret. Yes, very good. Very good. Now you will get answer sphenoid. Okay, you're going to get the marks. Sphenoid bone, it is a keystone bone. Why? Because it attaches with or it articulates with all other bone and it is kind of like a bat shaped bone, right? And it has a small depression called the sella turcica. Okay, very good. So answer is going to be option number 3. And students, hypothalamus. Okay, I didn't put the answer only. Sella turcica and hypothalamus. What is the stalk otherwise called as? What is the stalk otherwise called as? They can ask you any kind of question. Okay, don't expect that, okay, this question is put so only this question will be asked. No. What is the stalk called as? Come on. Quickly tell me in the chat box. Yes, bat shaped. Very good. Bat shaped. Very good. Yes, it is going to be called infundibulum. Very good. Infundibulum. Okay. So, this many things you have to remember here. Instead of stalk, they can give you infundibulum. Don't get confused. And pituitary gland is attached to the hypothalamus with a stalk. Okay. And they have the neurosecretory cells which are adding the secretions. Okay. So, that will give you 28 marks. Any student who has got 28? Yes, come on. Next question on the screen. Sclerites in cockroach are joined together by... Important question. Sclerites in cockroach are joined together by arthrodial membrane, Malpighian tubules, ganglia and phallomeres. Phallomeres. Yes, infundibulum. Very good. Okay. Come on. Arthrodial membrane. Excellent, students. Arthrodial membrane. Very good. So, what are they talking about? They're talking about sclerites means what? Tergites, sternites, your pleura. They're talking about that. So, that gives you 29. Okay, 29 marks. Very good. Moving on to the next question. Number of ommatidia in each compound eye of cockroach. Okay, each compound eye of cockroach. Come on, quickly in the chat box. Yes. Yes. Yes, Sweetie is telling to like the sessions. Okay, Ravi is telling. No shame you people have. Your friends are telling and you people are not listening. Okay. Yes, excellent, students. 2000 ommatidias. Now, please give me answer about the vision. What kind of a vision does cockroach have? Then I'll give you 1 mark. Okay, what kind of a vision? What kind of a sensitivity? Okay, what kind of a sensitivity? Quickly in the chats. Then you can take 1 mark. Okay, because 2000 is a simple question. So if you want 30, you should answer this 3 questions. 2000, vision and sensitivity. Oh, completed. My God. All the Kanjus have decided to be a part of it. Yes. Very good. Completed. Thank you, students. Yes, excellent, excellent. Mosaic vision, okay, and high sensitivity, okay, and low resolution. So now you can take 30 marks, okay, because that question is a very easy question. So 1 mark I cannot give for that. So yes, high sensitivity, low resolution. So now tell me your marks. 30. Sensitivity, don't know. Sensitivity is high sensitivity, okay? High sensitivity, low resolution. It does not have your DSLR, iPhone, Samsung capabilities to take a photo of you, okay? So sensitivity is going to be high, resolution is going to be low. Very good. Moving on to the next question. I hope all of you have calculated your marks. Yes, next question on the screen. The dithecus consists of dash microsporangia located at the corners, dash in each lobe, okay? Reading the dashes only I got confused. Yes, now let's make it 720. Exactly. That should be our target. Okay. Come on, students. Answer. The dithecus, okay, consists of microsporangia located at the corners. Okay. Dash in each lobe. Right? Yes. In school also I used to not like fill in the blanks. Why you tell? By the time I'm reading, the connection would have been lost, okay? Because that dash word is very important, but that word will be not there. So when I'm reading, I'm like, wait, what? What did I start reading on? So, yeah. So let's see what's the answer. Answer is going to be 4 and 2, right? Very good. So, dithecus consists of 4 microsporangia, okay, tetrasporangia, they are located at the corners. So, they have 2 theca, that is dithecus in each lobe. So, 2 is going to be the answer. Very, very good. Okay, very, very good. How much? 31. 31, how many of you got? Very good. 31, how many of you got? Yes, 4 microsporangia, 2 at each lobes, okay? Faster, let me see 31. How many of you got 31? Are you people playing with emojis? One is smiling, one is putting angry. Yes, very good. Very good. Okay. Yeah. You will not get 50, you will get 100 also. Okay. Yeah. Next one. Okay. Almost all the numbers are here. Okay. Almost all the numbers are here. Okay. Board-shaped mushroom gland, genital pouch, testis, 7th sternum, okay, 4th to 6th abdominal segment, 6 to 7, 8 to 9. If there is any student who is finding it difficult to solve this, that means you need to do that sheet which I told you, okay? Don't copy your friends and do, they also might not know it. I always tell this, okay? Don't do shortcut. Do everything. Shortcut, it seems. First only they're telling me, let's do shortcut. Shortcut. Oh, shortcut will work. Hi. Okay. Board shaped, 7th sternum, mushroom gland, 6th to 7th abdominal segment, right? Genital pouch, 8th to the 9th sternum, testis, 4th to 6th abdominal segment. What is the answer? What is the option C? Okay, very good. No, you cannot mark your answers in NEET question paper. It will be considered as malpractice. Okay, don't do all that. You have to do it in the OMR sheet. Yeah, maybe you can put a small dot or something, but you cannot like put that, okay, this is the answer. No. Okay, very good. 32. 32. Students, since you told this much, tell me ovary also. Tell me ovary also. Which segment ovary is present? Come on, quickly in the chat box. Okay, fill in the blanks and true or false. Okay. Thank you, Vaishnavi. Yes, students, ovary. Faster. All of you will know the ovary because I told you ovary is bigger than testis. So you started from an even number, you ended in an even number itself. Come on, who can answer? Let me see. 32, 31, very good. Yes, excellent, excellent. 2 to 6. Okay, 2 to 6. That is the second to the sixth segment. Second to the sixth segment. Excellent. Okay, moving on to the next question. 2 questions are there on the screen. Solve both. Okay, solve both. Sporopollen is absent in intine, exine, germ pore, A and B both. Okay, A and B both. Pollen grains are well preserved as fossils because of presence of intine, presence of germ pore, presence of sporopollen in all of these. Come on, it's I'll tell you there's something confusing here, so we will go into that. Okay. Students, we are not supposed to mark anything in the question paper. You people know it from school times, no? So that's the thing. Okay, some of you are confused. Okay, yes, very good. Students, you don't have just germ pore. Even intine doesn't have sporopollen. No, they're playing with your English. Okay, English knowledge is very, very less. Okay, very less you people have English knowledge. So it's going to be both A and B because intine also doesn't have sporopollen. Germ pore also, they do not have sporopollen. You understand the game with English. I told you, you people didn't request for an English class or else I would have taken. Pollen grains are well preserved because of the presence of sporopollen. So 32, 33, 34. 34, correct? 34. Yes, very good, okay? Those who did not understand the question, good job. Good job. Next question on the screen. Residual volume in lungs of a human is? Residual volume in lungs of a human is 500 ml, 3 to 4.5 liters, 800 ml, 1200 ml. Yes. Rough sheet they can provide you. Okay, rough sheet they can provide you. You cannot write anything on the question paper. Okay, you're not supposed to write anything. You can maybe put a small tick, okay, like a pencil mark, but you cannot write or solve all that in the question papers. Clear? Question paper, always be careful. It might be a trap. Okay, if someone tells you yes, you can write it on the question paper, it will be a trap. Yes, very good. It is going to be option number D. Excellent. Look at the speed at which the chats are moving. Very nice. 35 marks. Okay, 35, right? Yes. So 1200. Okay, students, label the following. Okay, full question you will get 1 mark. 2 marks I'll give you. Okay, so answer A first. Okay, then B, then C, then D, E and F. Come on, quickly in the chats. 1 by 1 answer. Okay, 1 by 1 answers. Generally, you have to practice the volumes. Okay, there's no way otherwise. See, it's very simple, students. First volume is going to be tidal volume. Normal inspiration, expiration, that is going to be 500 ml. Okay, sudden forced inspiration, that is going to be how much? 2500 to 3000 ml. Okay, by practice only we can remember. Expiratory reserve volume, that is ERV, is going to be how much? 1100 to 1200 or 1000 to 1100. Okay, 2 values are there actually in some books. If you see, 2 different values are there. And then you have residual volume. Okay, that is 1200. Only this much you have to remember. This 4 you have to remember at any cost. Okay, starting with A. Excellent. It is going to be called antipodals because I told you AC you bought in EMI. Okay, AC. AC you bought in EMI. We will come into that. B is going to be polar nuclei, you people are telling. Okay, very good. Polar nuclei. C, central cell. No one told her? Yeah, they told Abhishek. Okay, central cell. Then D, egg. E, synergids. Filiform. Okay, filiform apparatus. Correct? Yes, excellent. So, how many marks you want? 2 marks? Okay, plus 2. So, that will give you 37. Plus 37 in your pockets. Quickly tell me plus 37 in your pockets. Excellent. All of you have answered together. Very good. Very good. Some of you have answered scattered. No problem. Excellent choice. Moving forward. Next question on the screen. Oxygen dissociation curve is a hyperbolic, linear sigmoid. Okay. Hyper, linear, sigmoid. oxygen dissociation curve of hemoglobin is? What is it? Come on, students. Answer this also. Easy question. Perfect, perfect. It is going to be sigmoid. I told you people why the graph is sigmoid. Can anyone remember and tell me? I told you why we are not having a linear graph. Okay, why we are not having a linear graph. Instead, we are having a sigmoid curve. So I told you this. There's one reason for this to happen. What is that reason, students? Can anyone tell me what is the reason for the sigmoid curve? I told you this. I told you this is one special characteristic that oxygen has, that carbon dioxide will not have. Let me see who's going to answer first. Who is going to be the first person who remembers what I taught in class? Okay. There's no lag, dear. Come on. No, no, no. There's one word I used. Okay. Lag phase. Okay. Basaraj sir's class is over. It will replay after the entire session gets over. Okay. Now tell me this. No, not lag phase. One word I had used. One word I had used. What is that word? Something with. Okay. I think some of you take 1-2 seconds. Okay. No problem. To think and answer. Excellent. Excellent, Rani. Positive cooperativity of oxygen, not hemoglobin, oxygen. Okay, that is the mistake you have made. Yes, Rihanna, very good. Positive cooperativity. Okay, this is actually there in different publications of books. Okay, not there in your NCERT. But I told you positive cooperativity. Positive cooperativity means what? I told you one oxygen will enter. It will make sure that hemoglobin accepts the next oxygen as well. And this is how they dissociate. That is, saturation goes from 25 to 50, 50 to 75, 75 to 100. Understood? Understood? Positive cooperativity of oxygen, not hemoglobin. Excellent. Good job. So that's the answer. So sigmoid curve plus 36. Plus 36. Come on. How many of you got plus? Sorry, plus 37 will happen, no? 38. Okay, plus 38. So we have done 38 questions. Okay, 38 questions. Okay. Clear? Oh yeah, you people have this one, no? Yeah. Good job, good job. Moving on to the next question. The pollen grains in wind pollinated plants should be. This is important question, students. I'll tell you why it's important. Because such questions can be easy to score, at the same time easy to lose. Okay, it's a do or die kind of a question. Okay, do or die kind of a question. Clear? Yes. Now tell me the answer for this. The pollen grains in wind pollinated plants should be heavy and sticky, heavy and non-sticky, light and sticky, light and non-sticky. They are asking if you are, a plant who wants to do wind pollination, what will your pollen grains be like? Yes, think some of you are making careless mistakes. Excellent. The answer should be light and non-sticky. If they are sticky, then they are not going to separate from the anther and come. If they are non-sticky only, they will get scattered easily by wind. I know some of you will be thinking, okay, some other way, okay. So excellent. That is going to be light and non-sticky is going to be the answer. That is going to give you 39 marks. 39 questions we have done. So it's not just about marks, right? Yes. Exactly. 720 should be the target. Come on. Good job. Light and non-sticky. Okay. Because if they are sticky, they are not going to give out. They are not going to come out of the anther. No. After dehiscence also, they'll stick here. If you want it to go away, then it has to be light and non-sticky. Definitely, they're going to have sticky material to go and fall. But again, stigma has its own, capabilities and chemicals that will let the pollen grains fall on them. Excellent, students. Moving on to the next question, a big question. Okay. Tidal volume, inspiratory reserve volume, vital lung capacity and inspiratory capacity. TV plus IRV, ERV plus TV plus IRV, 500 ml, 2500 to 3000 ml. Okay. All of you can answer. Come on. Quickly answer. Tidal volume, I just now told you people. Inspiratory reserve volume also I told you. Vital capacity, again, capacities, you're going to have to remember the formulas. Okay, you have to remember the formulas. How many formulas you have? You have IRC, you have ERC, you have FRC, you have VC, you have TLC. This much formulas you have to remember, okay? And you have 5 capacities and 4 volumes. Clear? This also please write it in a small sticky note and keep it. 1 you people are telling, okay, let me see. A3, okay, A3, these 2 options can be striked out. 50-50, okay, I'm crorepati I'm playing, okay, 50-50. Phone a friend you want to do, you have to call me only. Inspiratory reserve volume is going to be 4, okay, so these 2 options are almost right. C, vital lung capacity is going to be option number 2, correct? Option number 2, okay, yes, it's going to be option number 1, correct? Good job, okay, very good. Yes, Priya, thank you. So 40 questions done. 40 questions done. Moving on to the next question on the screen. Why did I put 2? 1 is the answer. Okay, 1 is the answer. No. So once again, let me do 4. Correct. Okay. So D1. Yeah, this is the answer. Okay. D1 C2. Okay. Good job. Clear. Next question. Assertion reason question. Trachea, primary, secondary and tertiary bronchi is supported by incomplete cartilaginous rings. These rings of cartilage make the wall non-collapsible. Okay, you have to tell if assertion and reason are true. Okay, yes, go for it. Trachea, primary, secondary and tertiary bronchi is supported by incomplete cartilaginous ring. Reason, these rings of cartilage make the wall non-collapsible. Easy question, all of you can easily answer because you just now solved it. Yes, very good. Assertion and reason are true and reason is the correct explanation for assertion, okay, because they are talking about incomplete cartilaginous ring and they are continuing talking about these rings and mentioning about what the functions these people are doing. So that will give you 41 marks, okay. Next question on the screen. The cells of tapetum have, again, important question. Cells of tapetum, if you answer this, you're going to get 42. Quickly in the chats. Cells of tapetum, dense cytoplasm and single nucleus, dense cytoplasm and multinucleated condition, vacuolated cytoplasm and single nucleus, vacuolated cytoplasm and multinucleated condition. Come on students, answer. 40, 41, okay, very good. Hey, why are you telling A, B, C, D, everything you people are telling? Students, think of it this way. Tapetum is going to be the one who is going to do nourishment. If he wants to do nourishment, he should have a rich cytoplasm, right? If he wants to do nourishment, he needs to have something, okay? Yes, very good. The answer is going to be option number B, dense cytoplasm and multinucleated they are. That is the reason why they are able to provide the nourishment for the growing pollen grains. Remember that. If they do not have dense cytoplasm, how will they do the nourishment? Just think of it, right? Just think of it. Very good. Next question, 42. Anyone who got 42? I think many of you got 42, no? Next question on the screen. Pollen grains of 20% plants are released in 2-cell stage, released in 3-cell stage, non-functional, triploid. Come on, answer students. Pollen grains of 20% plants are released in 2-cell stage, released in 3-cell stage, non-functional, triploid. Easy question, you will be able to answer. Come on, quickly in the chats. Here, okay. Here they are talking about 20%, but see in your NCRT, they have mentioned 60% of the angiosperms are going to dehiscent at 2-cell stage. Okay, keeping the assumption 60%, 40%, the rest of them are going to go at 3-cell stage. Since they have not mentioned 40% and they have not mentioned 60%, we can definitely assume that they are talking about released in 3-cell stage, right? Yes or no? So 43 marks. Okay, 43 marks. Very good. Next question. Child death may occur in the marriage of Rh positive man and Rh positive woman, Rh positive man and Rh negative woman, Rh negative man and Rh negative woman, Rh negative man and Rh positive woman. Oh my god. Okay, so much to read. Come on. (43, 42), very good. C, no. 2, yes. RH positive man marries an RH negative woman. Okay, RH positive. So now if you're going to get married, you should check this also. Okay, the child, let's say the fetus, okay, is going to be RH positive. Then the erythroblastosis fetalis is going to happen, right? Yes or no? The mother is going to recognize the RH positive as an outside person, right? Like a foreign particle. And she's going to make antibodies against it, okay? Yes, very good. Yes, very good. So answer is going to be option number 2. Option number 2, that gives you 44 marks. 44 marks. Done? Very good, very good. Moving on to the next question. We are doing questions very fast. No wastage of time. How many male gametes are formed by pollen grains? How many male gametes are formed by pollen grains? Okay, students, tell me the extreme situations. Okay, don't tell me 2-cell stage, just tell me 3-cell stage. How many male gametes are formed by a pollen grain? No, it will not happen. Rani, then it will not be a problem if the child is RH positive. If the child becomes RH negative, again a problem will happen. Got it? It depends on if the compatibility is there or not. Okay? Yes? General situations we have seen where the mother is negative and the child is positive. Again, positive dominant, right? So that scenario will come. Yes. Answer is going to be option number 2. Alright? Answer is going to be option number 2. How much? 40. What's your marks, yeah? I can't remember numbers, students. In 2 seconds, I'll forget. 45. Okay, 45. Next question on the screen. The enzyme that degrades sporopollen is absent, deficient, mechanical, protective. Okay, mechanical, protective. Okay, again, see, remember this. Here we are talking about 2 in 3-cell stage. Okay, normally one pollen grain can make one male gamete also, you know, which will go and fuse with the egg. This is in 3-cell stage. Okay, we are talking about the extreme situations. Now answer this question. Excellent. Yes, very good. Very good. The answer is going to be absent. There is no enzyme that will degrade sporopollen. Okay, there is nothing that will degrade sporopollen. Next question on the screen. Alternation of which genes leads to cancer? It's a little hard question. So take your time and answer. Take your time and answer. Alternation of which genes lead to cancer? Alternation of which genes lead to cancer? Cell proliferation gene, that is proto-oncogenes, tumor suppressor genes, tumor virus genes, all of these. All of these. Come on. 46 is it? Okay. Okay, some of you are confused. I told you it's a little difficult question. Many students have made mistake over time. Students, the answer is going to be all of these. Okay, all these cells, that is this you have studied, tumor suppressor cells. From the term itself you understand, no, tumor suppressing cells, suppressor cells, tumor virus genes. Again, they also prevent from tumor formations. Okay, so all the 3, if they get any way mutated or any changes happen, that can lead to cancer. So some of you are saying A or D. I should choose the option, is it? Option D is going to be the answer. Okay, option D is going to be the answer. Many of you made mistake, so that will not give you 46 marks. Okay, you people are at 45. Some of you made right. Next question. In malignant tumor, the cells proliferate, grow rapidly and move to other parts of the body to form new tumors. This stage of disease is called as, that means that characteristic that is shown by malignant tumor where they have the ability to move from one place to another place and grow into another tumor, again move, spreading. That spreading is called as what? Yes, come on. Come on, come on, come on, come on. Very good, very good. Metastasis, metagenesis. What is it? Yes, you don't get confused. Metagenesis is alternation of generation. Alternation of generation, which is seen in your coelenterata. Okay. Now here metastasis is the condition. Clear to all of you? Okay. Metastasis. 47 marks. So this will be 48. Okay. 48 marks. We were supposed to do 50 questions, right? 50 questions. Next question on the screen. Which of the following conditions can be checked by Widal test or Widal test? Come on. Yes, metastasis. Genesis word comes for alternation of generation. Okay, always remember like this. Genesis, alternation. Okay, metastasis. That is where the growth is going to happen. Yes, very good. Excellent. Which of the following conditions can be checked by Weidel test or Vidal test? Yes, it is going to be typhoid. Typhoid. That gives you 49 marks. Okay, that will give you 49 marks. Next question. Which of the following is the correct sequence of steps in a PCR? Polymerase chain reaction. Okay, I have given you a trick to remember this, so you can write it. I mean, you can remember it faster. Come on, tell me the answer for this. Yes, typhoid. Very good. This is not A for Ammu ma'am. No, this is what? Yes. Excellent. Excellent. Answer is going to be denaturation, annealing and extension. Students, how you tell bay? Okay, like that you say day. Okay. So that gives you 50 marks. Our 50 questions are done, but we have some more questions. So let's do them. In a gene gun method used to introduce alien DNA into the host cells, microparticles of dash metals are used. Okay. In a gene gun method used to introduce alien DNA into host cell, microparticles of metals are used. What microparticles are used? Come on, quickly in the chat box. Excellent. Tungsten and gold. And students, please remember, they will ask you the same question with the question that gene gun method can be used in plants or animals. So that you have to remember it is plants. Okay, it is plants. So that gives you plus 51. Okay, plus 51. Moving on to the next question. DNA strands on a gel stained with ethidium bromide when viewed under UV radiation appears as dark red bands, bright blue bands, yellow bands, bright orange bands. Come on, quickly in the chats. Okay, and quickly mark your attendance, smash the like button as well. Okay, we should make 720 likes. Yes, excellent students. Bright orange bands, right? Because ethidium bromide, again, these people are going to be showing the strands in orange color. Okay, so bright orange color we are going to see. Otherwise, we will not be able to see the fragments that has run on a gel electrophoresis. Okay, that will run on a gel electrophoresis. Next question. Which one of the following equipment is essentially required for growing microbes on a large scale for industrial production of enzymes? Sludge digester, industrial oven, bioreactor, BOD incubator. BOD incubator. Easy, important questions though. Which one of the following equipment is essentially required for growing microbes on a large scale? On a large scale. Yes, excellent. Excellent. Answer is going to be bioreactor. Bioreactor. Okay. Again, they will have proper pH maintained. Okay. This we have done. Yes, answer this. Variation caused by mutation as proposed by Hugo de Veris. Okay. Random and directionless, small and directional, small and directionless, random and directional. Come on. Yes, bioreactor. Very good. Very good. Answer this, students. Variation caused by mutation. Okay. By whom? Hugo de Veris. Yes. If you have answered that, I also want you to tell me what did Hugo de Veris work on? On which plant did he work to come to this or to this conclusion that this is what is happening? Yes. Very good. Okay. Very good. The answer is going to be random and directionless. And the plant he worked on? Very nice. Very nice. Evening primrose. Okay, evening primrose. Okay, evening primrose. That gives you 54. One more question we will do and we will stop. Okay. Yes, this question. Which enzyme is responsible for the conversion of inactive fibrinogen to fibrin? Inactive fibrinogen to fibrin. Last question from my end. 55 questions we have done today. Can you believe 55 questions in a 1 hour span? Okay, don't worry, I'll share the PDF. In this PDF, there are some more questions. Okay, in the PDF, some more questions are there. I'll remove the answers from it. Okay, I'll just inform Shreyas sir. Yes, done. Okay, PDF. Oh, my phone. Okay, so sorry. Students, PDF will be shared on this Telegram channel. Let me just show you. See, a lot of questions are there more. Actually, we can do it in the next class. This Telegram channel, you will get the notes. At the same time, you will get it in Gopika Biology Official for all the biology, whichever chapters I've taken. All the notes will be available here and some additional informations as well. Okay, yes, got it. So thank you so much for joining. Okay, so now Shreyas sir is going to join, so I'm going to take a leave. I hope all of you enjoy the session. Please let us know in the comments how much you enjoy the session and are you confident in zoology as well. I think all of you did really well. So yes, please drop down your marks as well. So you would have got 55. Okay, 55 marks. How much you got? Please drop it down in the comments. I'm excited to see your marks as well. Thank you so much, students. Bye-bye. Bye-bye.
Good afternoon, my dear warriors. Welcome to the 200 plus questions. Very, very high important weightage questions coming up for all of you from the entire NEET Warriors team. It's time for the physics. I hope I'm audible and visible to all of you. Yes, looks like it. Looks like it. I'm seen at least. Please let me know, guys. How are you? And thank you for the super chats. I was just in the JE session just some time back, and thanks to Novo Kane for the super chat, and also to again Novo Kane. My God, thank you so much, yeah, for the super chats. And on JE channel, I remember in the last 3 days we got some 1,200 worth of super chats. It's not about the amount; it was just that gesture. Even if you had put one rupee super chat also, I would be very happy. But it's okay. By the way, don't worry, don't waste your money or don't spend your money, guys. All I ask you is just attend the classes, get a good rank. That's all we are asking for. And please leave a beautiful comment. It will not cost you anything. Just leave a beautiful comment like, yes, sir, we loved the session. Yes, sir, we enjoyed the session. Sir, thank you so much. That's it. One simple thing is the biggest super chat that I can get. Don't give any money. It's okay. Okay. And smash that like button down there. Thank you. Hello, Chintu. Hi, Renee. Hello, Meghna. Welcome, Dorothy. So let's begin with the class without wasting any time, because I know even Diksha Mam is eagerly waiting, and the exams are close by. Thank you guys for liking the video. Here comes the first question on your screen. Which of the following diagram represents Celsius versus Fahrenheit? Celsius on the Y, Fahrenheit on the x-axis. Now, degree Celsius is nothing but 5 by 9, and Fahrenheit degree minus 32. That is the formula, isn't it? Okay. So if you see, this is nothing but 5 by 9, F minus 5 by 9 multiplied by 32, and this is degree centigrade. Notice this is nothing but your y-axis. This is nothing but your x-axis. Y is mx plus c. So clearly the y-intercept is negative, and this thing is basically the slope is nothing but positive. Y-intercept is negative, slope is positive, so that means it will intersect somewhere negatively, but it will go up like that. So that graph is graph labeled A, which is option A. Yes. How many of you wrote option A? Quickly, let me see, guys. You should keep answering. Even if it is wrong, it's okay. Just keep answering. Yeah, perfect. Moving on to the next one. The graph over here, ABCA, as shown, what is the heat given? It's a cyclic process. ABCA is nothing but a cyclic process, and in a cyclic process, delta U is 0, and heat is nothing but the work, which is nothing but the area which is enclosed. So half into base multiplied by height is the area of the triangle. Half base is nothing but 1, height is nothing but 2, so it will be 1. So 1 joule is the answer, option A. How many of you gave it? See, that is how quickly. No, it is not. It is not B option. Why did you guys say B or D? B or D is not the right answer. So heat is work in case of cyclic process, because the delta U is 0 and work is area enclosed. So this is the area of the triangle. As you can see, this height is 4 minus 2, 4 minus 2, which is 2. And this is basically the base, which is 2 minus 1, which is 1. So that is why it is 1 joule. Yeah, perfect. Everyone, let's go to the next question. Assertion reasoning type. In an isothermal process, all the heat is supplied to the body is into internal. According to the first law of thermo, this is what it is. Yeah, first law of thermo, heat given is delta U plus W. So who am I? Am I a physics teacher or chemistry teacher? I'm a physics teacher, right? Diksha Mam teaches chemistry. I teach physics. So in physics, what is the law? Delta Q is delta U plus W. In chemistry, work done sign changes. So work done will come over here. Getting my point? So when Diksha Mam will be conducting the class, this work will come over here. So understand the sign convention changes. So heat supplied goes into internal energy change and work done. There is nothing wrong with it. And this is exactly the first law of thermodynamics. So reasoning is definitely correct. Okay. What about this? Isothermal process. In isothermal process, temperature is constant. So delta T is 0. So delta U is 0. So if delta U is 0, all the heat supplied is converted into, oh my god, all the heat goes into work. So it should have been work, not internal energy. It should have been work. So assertion is false and reasoning is correct. Assertion is false. D option. Wow, very good. Power Studios, Chintu, Sweetie, Deshila, Dhia, S Square. Very good, Manasvi. Hi, very good, Rian. Very good, Dr. Purple. Study logs. Awesome, awesome, perfect. Yes, that is the correct answer. The answer should have been work and not the other way around. Moving on to the next one. The PV diagram. We are taking up all high weightage because if you want to go from 300s to 500s and 600s, you have to tackle the high weightage topics in physics, my dear students. Okay. If you tackle the high weightage topics easily, you can boost your score to 100 marks. Moving on. 0, this. What is the work done in the process ABC? ABC. Okay, in the process A to B to C. It is nothing but the area of this particular semicircle. So what is the area of that semicircle? Area of that particular semicircle will be nothing but pi r square by 2. So pi is as it is, by 2 is as it is. What is the radius? So this is basically the radius. This is 2 units, which is diameter. So radius will be 1. So it is basically pi by 2. Now, whether it is plus pi by 2 or minus pi by 2, that you think and tell me whether it is plus pi by 2 or minus pi by 2. It is going clockwise, and clockwise is always positive, anti-clockwise is always negative. So clockwise, my dear students, so positive pi by 2 atm liter, not negative. See, negative is also there, chumma to confuse you. By the way, same question if Diksha Mam will conduct, she will say the answer is C. Understood? Because in chemistry, the sign convention is opposite. It is always work done on the system, whereas in physics, we take work done by the system. So expansions are positive, and in chemistry, contractions are positive. Moving on to the next one. Two rods of certain lengths, thermal conductivities are connected. If cross-sectional areas of two rods are equal, the equivalent thermal conductivity of the system is. Let's start drawing the two rods one after the other. One rod is here of length L and another is of length 2L. Okay. So there are your rods. All right. So this was L, this is 2L, this is 2K, this is K. So the total length will now be 3L. I don't know what the effective thermal conductivity will be. Because they are in series, I can say resistance 1 plus resistance 2 will be the equivalent resistance. Resistance is L by K into A. Next resistance will be 2L divided by K into A is total length 3L for the total resistance, K effective multiplied by A. If you want, cancel out all the unnecessary things like length and area so that it looks very easy to solve now. Okay, so this is 1 by 2K plus 2 by K is equal to 3 by KE. Therefore, 3 by KE will be 1 by 2 and this. So, LCM is 2K. So, it will be 1 plus 2 to the 4. So, it will be 5 by 2K. So, cross multiply. So, or basically take inverse. So, KE by 3 will be 2K by 5. So, therefore, KE will be 3 to the 6, 6K by 5. Therefore, KE will be 1.2 times of K. So, which is option A, I believe. Yes, that is option A. Perfect. Yeah. So, understood? Be careful when you read the statements also. Sometimes L2L, K2K, we will think, okay, it's in the same order, but K was for 2L and 2K was for L. So, it was exactly opposite, right? Shall we go ahead to the next question, my dear students? Wow, we have a match the following kind of question. So, think how are you going to solve this? The volume temperature graph are given, column 1, column 2, and choose the correct options. As you can see, A is matching with T most of the places, B is with PSTT, C with RTTP, D with TTTT. So no point solving for D. See, no point solving for T. Is that making sense? Okay, so T is with D, that is okay, fine. Now, volume of the gas is given. Gas is monoatomic, diatomic, pressure by PA by PB. Okay. So all these things have been asked. See, PV is equal to nRT. So volume by temperature will be nR by P. Volume by temperature, okay, is nothing but the slope of this graph. It is inversely proportional to P. So slope of A is more than slope of B. So that is why pressure of A will be less than pressure of B, because slope, whosoever is more, the pressure is going to be less. So pressure of A is less than pressure of B, and I think one of the options is like that. So PA divided by PB will be less than 1, will be less than 1. That is provided if the number of moles is given. But wait a minute, is the number of moles given? If n is a constant, or number of moles is for not constant, or if n1 is 1, n2 is 2, n1 is 5 moles, n2 is 3 moles, then we cannot exactly say. So this number of moles is not exactly given. If it is not given, we cannot really comment on this, because this is only true if both n1 equals to n2, which is not given to me, or na equals to nb, which is definitely not given. So cannot say anything should be the right answer. In fact, the number of moles is also not given. Gas B I cannot comment, gas A also I cannot comment. Nothing is given in the question. So is it like everything is TTTT? Oh my God, see, everything is TTTT. Very weird question, but imagine this question comes in the exam. This will take away majority of the students' marks. Majority of them will get negative marks here. None of them can be said, only because the number of moles of the gas is not known. You do not know anything about the moles of the gas. So yes, C option, my dear students. Many people were saying B option, but that will not be correct, because I do not know anything about the moles. And from a volume temperature, such kind of a graph, you cannot guess whether it is monoatomic or diatomic. That monoatomic, diatomic comes from, adiabatic curves. Adiabatic curves only. These are isobaric curves. Isobaric curves will never tell you whether it is monoatomic or diatomic so easily. So you have to go with TV raised to gamma minus 1 curves, which will be curved lines. From those curved lines, you can guess whether it is monoatomic or diatomic. So you really couldn't say anything even if moles were given. Yeah, cool. Let's go, guys, to the next question. 4 charges are there on the corners of a square. At the center of the square, what can you really comment about it? So these are the, this is the square. Charges are basically Q, minus Q, Q, and minus Q. So at the center, This negative Q and negative Q will give attractive fields. This positive Q will give repulsive fields. They will eventually cancel. So net electric field being a vector quantity will be 0. Is that right? What about potential? All the distances are same. So KQ by R, but again KQ by R will be there, and KQ by R will be there with negative sign, and KQ by R will be there with a negative sign. That will give me the net potential, which will be 0. All the charges are equidistant, R, and charges are equal but opposite in magnitude. So I think both will be 0. Oh, that's it. Option A, guys. How many of you wrote option A? Very good, Rian. Yes, Manasvi, whatever doubts, please ask. Please feel free to ask doubts. Yes, Emula, A option. Very good, MD Irfan, Nitya Sri. Awesome study log, sweetie. Very good. Power Studio, it's not B, it is A. Yes, Purni, it's option A. Very good. Moving on to the next question. The potential due to a charge distribution at the O will be how much? Potential. See, even if you take a small charge here, it is at the same distance r. So when you calculate the net potential, which will be K into that small charge which is there, delta Q, upon R, that R over here will be capital R. I will have to add all these potentials up. I will have to add all these potentials up to find the net potential at point O. This was just the potential due to a small charge, due to a small charge, due to a small charge delta Q. That potential will always be KQ divided by R. Is that right? So when you add them up, K by R is a constant. I will get delta Q summation, which is basically the total charge on it. And the total charge on it is nothing but charge per unit length into the length of the arc. Charge per unit length into the length of the arc. What is the value of K? It is 4 pi epsilon naught. This 1 by R. Lambda as it is. What is the length of the arc? It is 60 degree, right? So 2 pi R by 6, because it will be 1/6 of a complete circle. 60 is 1/6 of 360. So 2 pi R by 6. So R will cancel. 2 and 4 will also cancel. Pi and pi will also cancel. So you will just have lambda over here. 6 2s are 12. So 12 epsilon naught should be the answer. Yes, which is C option. Perfect. Yeah. K value, I put it as 1 by 4 pi epsilon naught. Everybody gets this. So this value of K is nothing but 1 divided by 4 pi epsilon naught. And the length of the arc, I just substituted it as total circumference by 6, because 60 degree is 1/6 of the circle. Yeah, option C. Very good. Many of you got it. Moving on to the next question. The charge on the 4 microfarad capacitor in the given circuit is charge on this guy. My God, this kind of problems, right? It's always lengthy, but it's okay. It's solvable. It's not that difficult also. So first of all, I don't even need these options. Yeah, just ignore them. Start combining the circuit into smaller circuit. So 1 and 5 together, these two together, they are in parallel. They are in parallel. So just add them. In capacitance, you just add it. It becomes 6 microfarad. Now, what you do is redraw the circuit. So just imagine this to be 4 microfarad and this to be basically 6 microfarad. That is in parallel with your 3 microfarad, and that is in parallel to this battery over here of 10 volts. Now this 6 and 4 are in series. Yes, this 6 and 4, they are in series. So let's do that. What will be the effective combination of both of them? By the way, this is just 3 microfarad and their combination is over here like this, 10 volts. Yeah, just do the math, guys. It will be nothing but 6 multiplied by 4 by 6 plus 4, which is 24 by 10, which is 12 by 5, which is 12 by 5 microfarad. Yeah, 6 and 4 will be 12 by 5. So that's it. Just put over here 12 by 5 microfarad. Now these two together are in parallel. So eventually what will happen is they will become only one single capacitor with one single battery over here of 10 volts. So this capacitance will be how much? Just add them. So 3 plus 12 by 5. 5 3 is a 15 plus 12 by 5, which is 27 by 5. So 27 by 5 microfarad. So 27 by 5 microfarad. 27 by 5 microfarad. Is that right, everyone? Very good. Now what to do? Now what to do? Observe this carefully. Now what to do? Question says find the charge on the 4 microfarad. You have to go exactly the reverse. You combined, reduced it, and brought it till here, right? So now you go exactly the reverse. So how much will be the charge on this guy? You think how much will be the charge on this guy? CV. C is 27 by 5 into V, which is 10 by 5 is 2. So 27 into 2 is 54 microcoulomb. That charge will now get distributed over here and then distributed over here and all of that. So first of all, because this was the charge on this guy, 54 microcoulomb, actually we didn't even have to do this. We could have just started over here only. How much charge will be there on this guy? Let's say I call it Q1. It will be capacitance, which is 12 by 5 into voltage, which is nothing but 10. So 10 and 5 goes 2 times. So this will be nothing but 24 microcoulomb. So if 24 is here, obviously the charge on the other guy, Q2, will be 54 minus 24. 54 minus 24, which is nothing but 30 microcoulomb on this guy. Now this charge on this 12 by 5 will get distributed here equally. Yes, this charge will get distributed equally because they are in series. So in series, it is always the same. So the answer will be 24 microcoulomb. So on 4 microfarad capacitor, the charge is 4 microcoulomb. That is the answer. Actually, there was no need to find this out also. At this step only, we could have stopped. The voltage will be same on this and this. So charge on this guy is C into V, capacitance into voltage. And this same charge will be there on both of them. Directly, we could have told it at this point. So answer is 24. Just check this out. Yeah, B option is the correct answer. How many of you gave it? Very good, Manasvi Chintu. Awesome, Janani. Murugan. Very good, Sadhu. Very good, Power Studios. Yes. How to solve this question in the short duration? I'll tell you what. There is no need to solve this part. Okay, even this part was not needed. Even this part you could have avoided. Just imagine mentally, okay, these two are in parallel. So 6 and 4 in series. So it will be 12 by 5. So 12 by 5 will have the voltage of 10 volts. So how much is the charge? 24. Whatever charge is over here will also be there on both of them. Directly you can give the answer. But I have solved it till the end just to show you that in case we needed to find anything in the entire problem, we will probably have to go till the end. But certain things can be solved in a much shorter way. Like for example, this one was little bit quicker. You did not have to get this. Is that okay? Perfect. Yeah. Right. So just if you had to solve it in short, there was no need for solving this part. You need not have to solve these things. This was not even needed. Even this obviously was not needed. Now it looks short, isn't it? Now this is basically the short way, right? Okay. So certain problems will definitely take time. Certain problems will take less time. Wherever you take more time, you have to make it up with the easier problems. Next one, two different isotherms represent pressure volume, okay, of the same ideal gas are shown and of the masses M1 and M2. Then what can you say? This is very interesting, guys. Okay, two different isotherms of the pressure volume are given, okay, at a given temperature for an ideal gas. So we all know PV is equal to nRT if temperature is same, R is same, pressure into volume is directly proportional to N. Usually what used to happen if temperature increases, see, under normal circumstances, what used to happen is you had pressure volume graph. If, let's say, the temperature was lower versus the temperature was higher, the graph when the temperature was higher used to shift on the top. That means nRT will be higher. Same thing should happen here. If n is small versus n is big, what should happen? If n is small, it should be over here. Okay, and if n is big, moles are big, it will be over here. So less moles means less mass, more moles means more mass. So m2 will be more than m1. Directly you can say it. So the product of pressure into volume, if it is higher, that means the graph will shift more towards this side. If the product is less, that means moles are less, the graph will shift more closer towards the origin. That's why C option. Yeah, very direct, guys. Very good. Let's go to the next question on your screen. A conducting circular loop carries some current. It is placed in a magnetic field such that B is perpendicular to the plane. What is the magnetic force? I mean, you don't even have to think about it. Whenever you have any loop, doesn't matter. And if you find that the magnetic field is present, whether parallel, perpendicular, doesn't even matter actually. Even if it is at an angle, it is okay. But if this magnetic field is uniform, and if this is a loop, 100% the net force on that loop will be 0, irrespective of the shape, size, orientation. So all the other data does not matter, it is 0. Torque will not be 0, but the torque will be there. M cross B, torque will be there. Keep that in mind. Torque may or may not be there, that depends on the orientation. Next question, guys, coming up on your screen. Wow, we have something related to electric field, magnetic field, and the forces. Just think about it. Sir, is it enough to solve 2019 to 24 PYQs physics? Can I get 100 marks by this only? No, Arisha, that is not enough to get 100 marks in physics, 100%, I'll tell you, because there are so many type of questions which can come even beyond that. So apart from that, so in the days which are there, apart from that, give the mock test. That will be good enough to give you 100 marks. Okay, just don't depend on PYQs, because not all PYQs will repeat. Some models will repeat. Number two, you cannot just be dependent on that. So obviously you need little bit extra edge, that is at least give the model mock test, which we are doing here, plus the NEETpedia. I hope you know this. I have given you all these extra material for free of cost. I had made a special video, guys, on it. I don't know whether you guys saw it. NEET Revise-a-thon. High speed revision of all the chapters. We have kept it right over here in this particular PDF. I can't go up or down for some reason. Maybe my touch screen has a problem. It is there, guys. All India mock test series are there in this. Okay, NEETpedia is there in this, where you have all the revision book, physics revision book. For example, if you click on this, all the important formulas are there. You can just go through it. This PDF is so nice, detail also, and everything is there. Free of cost it is there. It is only there for a limited time, because obviously NEET exam is there around the corner. We want to help all of you in this time of need. So make use of this. All these test series are also free of cost. Champions test series. So give this. So give all these test series. 100% you will get more than 100 marks. You might even get 120 marks. Okay, cool. Let's go, guys. Charge particle enters a region, orthogonal fields perpendicular, then what can you say about the velocity? Okay. Yeah, what happens is, this is crossed field concept. The velocity will be like this, and the electric field, for example, it is like this. Then the magnetic field is in this direction. So E cross B or B cross E? E cross B is giving you V. Yes, so B and B cross E, and that C option is eliminated. So as you can see, electric field cross magnetic field. Magnetic field is inside and electric field is over here. Yeah, electric field is over here. If you do E cross B, the direction is coming out over here. So this is the direction. If you do the cross product of electric field cross the magnetic field, which is definitely parallel to velocity. So either B option or D option. Now, how do I choose that? We know the magnitude of the speed is E by B. We know the magnitude of the speed is E by B. So deliberately, if I put B over here, it will be E by B square. B will cancel. Now it makes sense. Now it makes sense that if I had V over here, I can put E cross B by B square. So E cross B by B square is B option. Can you see that? I just manipulated the equation, guys. Yes, I just manipulated the equation. So B option is the right answer. Directly you can say it. Correct? Okay, very good. Let's move. There is a air capacitor. Okay, when it is filled with a dielectric medium, then half the space, the capacitance becomes 2 by C, the value of K is. These kind of questions actually boil down to, you have now two capacitors like this. Think of it like that. If you take half of this, area becomes half. Epsilon naught A by D, area and capacitance are proportional. So this will be C by 2. This will be also C by 2. Why? Because capacitance formula is epsilon naught multiplied by area divided by distance between the plates. So it was directly proportional to the area. So half the area taken, so C by 2. But out of that also, you have filled this particular capacitance with some dielectric medium. So it will become K times. Now this combination is in parallel, guys. This is a parallel combination. Can you see that? This particular combination is a parallel combination. So C effective is C1 plus C2. The total capacitance is found out to be 2C. C1 is nothing but C by 2. The other capacitance is KC by 2. Okay. CC cancels. So 2 is equal to half plus K by 2. So K by 2 is 2 minus half, which is 1.5. So K will be 2 multiplied by 1.5, which is basically 3. So 3 is C option. Yes, directly you can get it. So when it is like this, right? So just think one of the terminal is over here, one of the terminal is over here. The charge will get over here also and here also. So it is basically in parallel. So when you draw an equivalent circuit, show two capacitors in parallel. When it was like this, say for example, one of the terminal is here and this is here, this is like this will be series. This is one after the other. The charge will have to go from this capacitor to this capacitor one after the other. That is series. When it is sideways like that, that is parallel. Okay, don't get confused. Many people get confused, sir, whether it is parallel or series. Okay, moving on. The reading of the ideal ammeter shown in the network at steady state. Ideal ammeter network, okay, at steady state. First of all, why is this even capacitor given? It's completely useless. It is going to block the current. So the current won't even go there. Whatever current is there from the battery will just go in this part of the circuit. And the current will be total voltage divided by total resistance. Total voltage 30, total resistance 10 plus 5, which is 15. 30 by 15 is basically 2. So 2 amperes is the current. What is the reading of the ammeter? But why have they given it in voltage? I think the misprint is there over here. I'll just correct it. Yeah. So I was just wondering why it is given in voltage. This was supposed to be amperes, guys, not volts. Because current reading is asked in the ammeter. So I'll just put ampere over here. Okay, this was also ampere, and ampere. Okay, perfect. So 2 amperes is the answer. How many of you gave it? Very good. C option. Okay, very good. Mother switch into Renine, Levy. Good practice happening, guys, at this point of time. Very good. Keep it up. Keep it up. Moving on to the next question. For a given conductor, the drift speed of the electrons at cross section 1, 2, and 3 are. How is drift speed related to the area of cross section? Current is NVDR. N, I, and E are constant. So drift speed is inversely proportional to area because the current will be the same no matter what the width is. Same charges will have to go through every cross section. So out of this, who is the least? Out of this, who is the least and who is the most? So A3 is the highest, then comes A1, then comes A2. Drift speed will be exactly opposite. So drift speed in 3 will be the least, then it will be 1, then it will be 2. So V3 will be the least over here, then V1, then V2. V3, then V1, then V2, which is exactly this particular option. Correct? C option? No, 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 it is not D. Sneha, be careful. I think you made a small mistake there. You have to do it exactly reverse. 3 is the highest, 2 is the smallest. So 3 will have the smallest speed and 2 will have the highest drift speed. Current is equal to NVIDIA. Yes, current is equal to NVIDIA formula. Cool. Let's go to the next question. Current of 2 amperes is flowing through a resistor when connected across a battery. The same battery supplies a current of 0.5 when connected to the internal resistance. This kind of questions are also very, very common. All right, so how do we do this? The notes of the marathon will be available as PDF after the session is over. Link will be there in the description as well as in the pinned comment section. Okay, fair enough. What is there? Just think of a battery, internal resistance like this, external resistance. This is R, this is E, this is capital R, this is current. So this current will be total EMF by R plus R. So if you look at it the first time, the current is 2 amperes, EMF I don't know, external resistance 2, internal resistance don't know. Second time the current is 0.5, EMF I don't know, external resistance 9 and this is plus R. So obviously better to divide both of them. So divide both of them. So 2 divided by 0.5 will be, this will be E divided by 2 plus R. This will be E divided by 9 plus R. E will cancel. 2 divided by 0.5 is nothing but 4. And 9 plus R will go on the top. And here we'll have 2 plus R. Cross multiply. So it will be 8 plus 4R will be 9 plus R. Okay, take R over here. So 4R minus R. 9 minus 8 will come over here. So this is 1. So this is 3 times of R. This is 3 times of R. So R will be 1 by 3 ohms. 1 by 3 ohms, which is A option. Yes, it is the correct answer. No, it is not B. It is A option. Power Studios is correct. Manasvi is correct. Ravindra, I think you made a mistake. Yes, Levi, the option A is the right answer. Very good. Let's go to the next one coming up on your screen. The de Broglie wavelength of an electron moving with kinetic energy of 144. This is standard question, guys, standard formula. What is it? 1.227 divided by root of the potential difference. This is in nanometers. Okay. So if the kinetic energy is this much, the voltage by which it was accelerated was 144 volts. 144 volts, because the kinetic energy is the charge multiplied by the voltage difference. The charge of the electron is E. The charge of the electron is obviously E. So that's why kinetic energy was 144 electron volts. So voltage we got. So substitute it over here. So this will be, what is it? 12.27 into 10 to the power minus 1. Nano is 10 to the power minus 9. Okay, deliberately I put minus 1 because I'm seeing 12 over here and I've shifted a decimal. This is root of 144. Root of 144 and 12 approximately the same. So I think it will be 1 point something and this will be 10 to the power minus 10 meter. So almost 1. So into 10 to the power minus 10, if you want it in nanometers, then just leave that as it is. So this will be 10 raised to minus 1 into 10 to the power minus 9. So 10 to the power minus 9 is nanometers. So if you want 10 raised to minus 1 in the answer, so if you shift it, how many times? If you write it 102, if maybe this was 1.02, if you write it 102, then it will become 10 to the power, you have shifted it 2 decimals, minus 3. 10 to the power minus 3. 102, 10 to the power minus 3. Is that right? Is that making sense now? See guys, what I've just done. 10 to the power minus 9, just keep it as it is. If you want, don't even bother writing this because everything was in nanometers. Just keep nanometers only. It's okay. Just keep nanometers. 12 and root 144 is the same thing, almost. This is 12. So 12.27 by 12 will be almost like 1.02 or something like that, say. So if this is raised to -1, shift the decimal 2 places. So this will become negative power 3. So I think option A should be the right one. Yes, perfect. Yeah, cool guys. Very nice. Moving on to the next question on your screen. The photoelectric emission process for a metal of work function 1.8, kinetic energy is this much, the corresponding stopping potential is. Guys, this question is direct. Already the kinetic energy is mentioned. So the kinetic energy, maximum kinetic energy is E times the stopping potential. This is useless data. So 0.5 EV is EVS. So stopping potential is 0.5 volts directly. 0.5 volts. Yep, C option. What is the use of this? Completely useless data. Chumma, to confuse you, they have given it unnecessarily. Yeah, is that right? Correcto. Let's go guys to the next question on your screen. There is some kind of street lamp, yellow light, assuming 25% efficient in electrical energy to light energy. So the number of photons of yellow light will be. First of all, because it is 25% efficient, the power of light, the power of the actual light source, 25%, that is 25 by 100 of 200, which is 1/4 of 200, which is basically 50 watts of energy will be going in light. Now we all know power is nothing but energy which is coming per unit time, but there will be many photons. So each photon will have some energy EP. So divided by time. Now they are asking how many photons are emitted per unit time. So N by T. So N by T will be P divided by EP, and P divided by EP means this is P divided by Hc by lambda. So now substitute everything. Power is 50, wavelength is 0.6 micro, so 10 to the power minus 6, whole thing divided by 6.6 into 10 to the power minus 34, and lastly speed of light, which is 3 into 10 to the power 8. Yeah, that is what it should be. So just see how much answer you are getting. Almost like 1.5 you should get. Almost like 1.5 you should get. Makes sense? Just check this out. Close enough to 1.5. And I'll tell you an easy way to solve this. See, I'll tell you an easy way to solve this. See only the numbers. Ignore the decimals, zeros, and the powers of 10. You have 5 over here. You have 6 over here. You have 6.6 over here. And you have 3 over here. The 6 and 6.6 will become 1.1. So what is 5 by 3, guys? What is 5 by 3? It is basically 1.6, 1.66, and this is 1.1. So this is 16 by 11. Almost approximately how much will it be? Almost how much will it be? Almost 1.5. So the answer will be 1.5 into 10 to the power something. So see, wherever there is 15 number, directly you can mark it as option A. So that is the calculation trick where you don't have to break your head, ignoring the powers of 10, decimals and zeros. Making sense? Yeah, very good. Moving on to the next question. The wavelength of the electron and the photon of the same energy are related by, of the same energy, electron and photon. Well, energy of a photon is hc by lambda. For an electron, what is that energy? Lambda E by lambda is nothing but h by root of 2m into E. Oh, so just substitute it over here, h by root of 2m into E. Instead of E, put over here hc by lambda p. Okay, so squaring both sides, we will get lambda E square is equal to h square divided by 2m, and we will have hc divided by lambda p. So I think what you can also write is lambda E square is h square by 2m. This hc will stay over here. This hc will stay over here, and this lambda p will go on the top. As you can see, the square of the wavelength of electron is proportional to lambda p. Square of the wavelength of the electron is proportional to lambda p, which is C option. So directly we could get it. Not very difficult. Yeah, C option. This question model has been repeated many times in many competitive exams. Moving on. What is the shortest wavelength present in the Bastian series of the spectral lines? Direct formula based, guys, not at all difficult. Yeah, direct formula based, n1 square and n2 square. So what is the value of n1 and n2 that you will put? Bastian. Shortest wavelength. See, when the wavelength is shortest, lambda is shortest, then the energy difference will be the highest. So energy difference will be the highest, means Lyman-Balmer-Bastian. So 1 will be 3, n2 will be infinity. N2 will be infinity. And the Rydberg constant everybody knows. So solving this, you will get the value of lambda. Solving this, you will get the value of lambda. Yeah, 3 and infinity. Direct formula based. Cool? Right. Let's go up to the next question. The answer was 820 nanometers. Yeah. So you'll get over here 820 nanometers after directly substituting the values. Nothing great about the question, guys. Okay. Yeah. So this question just got repeated 2 times, I think. My bad. Next question. An electron is accelerated from the rest through a potential difference of some volts. The de Broglie wavelength is this much. What is the voltage? Again, same formula, guys. De Broglie wavelength for an electron accelerated through a voltage difference is 1.22. Okay, you can just put 1.227 into, or if you just want put nanometers divided by the voltage difference. Okay, so the wavelength is already given. It is 1.227 into 10 to the power minus 2 nanometers. This is 1.227 nanometers. This is delta V. Nanometers, 1.227 gets cancelled. So 10 to the power minus 2 is 1 by root delta V. So squaring both sides, it is 10 to the power minus 4 is 1 by delta V. So therefore, basically 10 to the power 4 is nothing but delta V. So delta V is 10 to the power 4 volts, which is B option. Directly you can get it. So this formula is only valid for electrons, and you should remember this value because it will simplify your calculations by a big deal. Cool? Let's go, guys. Next one. The graph between energy and its de Broglie wavelength will be. Okay. So we all know lambda is nothing but Planck's constant by root of 2 times mass times energy. So h by root 2m is a constant. So the power of E when taken on the top numerator, it will be e raised to negative half. Now take logarithms on both sides. So log of lambda will be nothing but log of nothing but h by root of 2m, and this is e raised to negative half. Now log of a into b is log of a plus log of b. So this will be nothing but log of, come man, h by root of 2m plus log of e raised to negative half. That's it. Yeah, about it. Okay. So this will be nothing but log of h by root 2m. Whenever you have power, log of x raised to m is m into log of x. So log of x raised to m is m into log of x. So this will become nothing but negative then negative half log of e. Okay. So this is nothing but log of lambda. So now treat log of lambda as your y-axis and treat log of e as your x-axis. Clearly you will see now that this negative half is nothing but your slope, and this thing over here is nothing but your c. So y is mx plus c. Intercept is positive but with a negative slope. So C option. Can you see? This is nothing but the y-intercept, which is nothing but your c, and the slope is definitely negative. Slope is definitely negative. So C option. Correct? So direct comparison with straight line equation. Moving on to the next one. Photons, they are deflected by electric and magnetic fields. 100% wrong. How can they be deflected by electric and magnetic fields? They are chargeless particles. Yeah. So they do not have any magnetic property. Yeah. Okay. All right. Then wavelength of a photon changes as the medium changes. Yes, this is correct, guys. I mean, we know that C1 by C2 is lambda 1 by lambda 2? Definitely. Number of photons are conserved in electron-photon collision? Definitely not. They are not conserved because they can be absorbed or they can be emitted also. Yeah, correct. So 1 and 3 are wrong, and the question says choose the incorrect option. So 1 and 3. 1 and 3 means C option. So C option will be the correct answer, which talks about the incorrect options. Yeah. So be careful. Many times people will think, oh, only number 2 is correct. So they will immediately mark a D option. Be careful. Many people mark D also. I can see it. See, Ninja, Gemma, yeah, Manasvi. So many of you mark D option, but D is not correct because the question says which is incorrect. So 1 and 3 are incorrect. See, this kind of questions you will feel very bad after coming out of the examination hall that you did it properly, and just because you read it wrong, you have got negative marks. So be very, very careful. Thank you. Thank you so much for all those likes, guys. And so cool to see your love on the channel. Thank you so much, guys. Means a lot. Oh, thank you. Thank you for 600 likes. Hopefully, this will reach 1K likes very soon. It means a lot when you show your love for all the teachers out here. And this is a channel which we created specially, for all the students who want to learn in English. And we have Hindi channels, but uh, many people tried creating English channels, but obviously you need good quality teachers. And many times South Indian students from Northeast and many metro cities were neglected. So thank you so much for showing all your love for the quality content that we give. Moving on to the next one. The graph between the square root of the frequency of a specific line characteristic spectrum of X-rays and the atomic number of the target will be. This is Moseley's law. This is Moseley's law. Directly you should be able to give it, guys. Yeah, directly you should be able to give it. Yeah, B option. Very good. Moseley's law. Root of nu is proportional to Z. Oh, by the way, we are planning to come to Tamil Nadu after your NEET exams are over. Would you love to meet us or no? And right now we are planning 11th of May in Chennai, Tamil Nadu. If you want to meet Gopika ma'am, Baswa sir, Diksha ma'am, Capto, then let us know after the session is over. Yes, sir, we'll be ready to meet you. You'll be like, sir, what will you do after our exams? My dear, your counseling. Yeah, whether to take this college, that college, whether to take a drop, not drop, and plus your juniors will be also there, no? So please get your juniors also along. It will be very amazing to get your batchmates, your juniors, your classmates, or your relatives, your cousins, your friends, maybe somebody whom you know and needs help, everything. So you will get to meet all of us. So we have been traveling to many places, Bangalore, Pune, and then last year also we did workshops in Chennai and Madurai, Trichy, so many places. So now is the time. You work really hard, and we'll be very delighted to meet all of you, irrespective of whether you score good or bad. Don't worry about it. It's not like, sir, I've scored bad. Can I come and meet you? I'd feel very bad. In fact, then I would like to meet you more. That because you need more help then, right? And if you've scored well, then I would be even eager to click pictures with all of you. I'm pretty sure it will be a great time. Okay, so see you very soon, guys, in Chennai and Hyderabad. Oh, don't worry, we are coming there to Hyderabad, and most likely 17th May is the plan. 11th May in Chennai and Hyderabad, and on 17th of May. Okay, that's the tentative plan. I'll keep you guys posted about it. Yeah, Madurai and Trichy also you want? Okay, fine. If Madurai, Trichy, more comments come, we'll make a form and then we'll make probably plan more visits. Madurai and Trichy, yeah. I want to probably do a road trip from Bangalore after NEET exam is over and all these exams, J once everything is over. Like from Bangalore, just drive it down from Madurai, then go to Trichy, go to Kanyakumari, and on the way back maybe come via Pondicherry, Chennai, and then back to Bangalore. It's one it's like a dream trip for me. Yeah, go to Rameshwaram and all of that. Let's see how it goes because my time is very constrained and limited, but someday I would love to do that. Oh, Goa, Reni, you are from Goa. Goa to keep visiting, man. Yeah. So following circuit acts like which kind of gate? Which kind of gate does it act like? Goa, I would love to visit. Oh, Gopika ma'am is already in. Gopika ma'am is the first person to register for the event already. So we have one enrollment already. So let's see how many enrollments we cross. Yeah, Kashmir. Oh, definitely. Salem though. See guys, if I visit Salem, no, the problem is average velocity will be zero then. Why? Because Shreyas sir was born in Salem. So what will happen? Displacement. Initial position and final position will be same. So displacement will be zero. So average velocity will become zero. That was a very bad joke, but still. Anyways, coming back. What is this, guys? Whenever you have a NAND gate and you short the two terminals, what will happen? It will always behave like a NOT gate. That is the trick for solving these questions. Not, 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 not. Oh my God, so many knots over here. Okay, so this is, oh, why did they give the answer? This was not supposed to be given, guys. Okay, not supposed to be printed in the question. By mistake, they have given. Okay, fine. So if this is A, this will be A bar. If this is B, this will be B bar. This is a NAND gate with a NOT in front. So these two together will just become a AND gate because it's a NAND plus, think about it, it is a NAND plus NOT. So it will just become a AND gate. It will just become a AND gate. So that's it, guys. So A bar and B bar will be what? Use De Morgan's principle. Dot will become plus. The bars will combine together. So what is this plus? It is a OR gate. So it is now essentially becoming a NOR gate. A NOR gate. So NOR gate is option A, I believe. Yes, that is the right answer. Perfect. Very good. Oh, Diksha Mam is also in. Oh, so two entries already. Where is Baswa sir? Baswa sir is missing. I think Baswa sir will be late. He will come on his bike. Baswa sir will be the last person to register. If Baswa sir registers after the students, then no food for Baswa sir. Yeah. So let's move, guys, to the next question coming up on your screen. Which circuit will not show the current in the ammeter? Which circuit will not show the current in the ammeter? That means somehow there should be some kind of reverse bias because I can see some diodes over here. Now, if you see this circuit, this is forward bias, this is forward bias. Obviously, the current will flow in this. Here, 100% the current will flow through both the branches. So this is also fine. Here, one of them is reverse bias, another is forward bias. So the current will come. It will not go over here, but it will go through here and it will come in the circuit. Here, this itself will block it. So this is in reverse bias. So no current will flow. So no current in the ammeter. So option A. So which will not show current? This will not show current. It's in reverse bias. It's out of question. Yeah, correct. Very good. Okay, let's go to the next question coming up on your screen. Transistor, oops, sorry guys, transistor questions are not supposed to be asked. They are out of the syllabus. My bad. Yeah. Okay, yeah, option A was correct for that previous one. Very good. Moving on to the next question. From a circular disc, 9M, a small disc of radius r by 3 is removed concentrically. Thankfully, it is concentric, or else it will be a much complex question. The moment of inertia of the remaining disc about the axis perpendicular to it will be how much? Okay. So, see, my dear students, this is your disc. It has a mass of 9m, of course. This is your axis, and radius is also given to be r. So, from that, what you have done, you have taken out a smaller disc whose mass is basically m. Is that right? And radius is basically r by 3. Okay, let's think what is the moment of inertia of that big disk. So it is half, because it is mr square by 2, right? Half into 9m into r square. What is the moment of inertia of that small disk? It is half into mass into whatever is r. So r by 3 whole square. So the question is now, what is the moment of inertia of the remaining part? So the moment of inertia of that remaining part of that particular disk. Okay. So that moment of inertia of that remaining part will be this minus this. So 9 mr square by 2 minus this one mr square. 3 square is 9, 9 2s are 18. So minus 18. Is that right, everyone? Perfecto. So LCM is basically 18. So this will become 9 times. So this will become 9 into 9, which is 81 mr square minus mr square. Okay, so it is 80 mr square by 18 divided by 2. So that will be 40 mr square by 9. So 40 mr square by 9 is option number A. Yes, that is the right answer. Can you see that? Many students said A, some of you said B. Yeah. Yeah, transistors are deleted, my dear. Yeah, transistors are deleted. They're not there. Only diodes, only little diodes. Even in NCERT, transistors are deleted from the new NCERT. Cool. Let's go, guys. In other exams, it could be there, like local common entrance tests for state governments. Some places it is there. Thin disk of mass m radius r is rotating about its axis with some angular speed. Two objects of mass m are attached gently to the opposite diameter. The ring now rotates with. This kind of questions based on completely angular momentum conservation. Angular momentum conservation, which is I1 omega 1 is I2 omega 2. So if it's a ring of mass m and radius r, initial moment of inertia is mr square. Omega, I think, is given, let's say omega. Okay. Next, because the ring will itself be there plus two additional masses which are gently attached, each mass will have a moment of inertia of mr square. So the new moment of inertia will be this whole thing, and this will be the new angular speed. So as you can see, this is nothing but mr square plus 2 times mr square, and this is omega 2. But the best part is this r square and this r square will cancel, and I am left with m omega is m plus 2m into omega 2. So omega 2 will be m omega divided by m plus 2m. M omega divided by m plus 2m, which is the D option right over here. Is that correct? So based on angular momentum conservation, and when you add two extra masses, they will have extra inertias, mr square. So just add these two terms over here. No, not C option. It is a D option. Be careful. Yes, very good, Power Studios. Very good, my dear. Very good. Okay. So I think we'll go to the last few questions now coming up on the screen. After this, Diksha Mam will come in for chemistry. Force is acting at a point. What is the value of alpha where which angular momentum is conserved? Angular momentum is conserved. That means torque should be 0. So their cross product should be 0. Is that right? Yeah, the cross product should be 0. So let's start writing down everything. So I cap, J cap, K cap, R cross F. R is basically 2, -6 and -12. Force alpha, 3 and 6. Determinant method, my dear students. Yeah, use the determinant method. So this is basically I cap. Something will be there. Minus will come then, not plus. And then finally again plus will come over here. That is how the determinant method works. Okay. Now for I cap, ignore this column, ignore this row. Just look at these four. Just multiply this and multiply these two and subtract them. -6 into 6 is -36. 12 into 3 minus 12 into 3, 36. So minus 36. Okay, fair enough. For J cap, forget this column, forget this row. So 2 into 6, 12. Minus 12 into alpha, minus 12 alpha, but again put a minus sign. Then for K cap, forget this column, forget this row. Just look at these two numbers. 2 3s are 6. Minus 6 into alpha, minus 6 alpha, and then put a minus sign over here. Okay. So the angular momentum is conserved. Have they missed the signs or something? Because yeah, no, this is fine only. Yeah, minus plus. So this will become basically 0 I cap. Now over here, this will become 6 plus 12 alpha minus J cap, and this will also become nothing but minus is plus. So 6 plus 6 alpha, and this is nothing but K cap. Just check this out. Is this what it is? Yeah. Perfecto. Have we missed anything? Just check it. 2 minus 6 minus 12, alpha 3 and 6. So this is minus 6 into 6, 12 into 3, okay. For J, 2 6 are 12 and this is minus 12 alpha. For K, 2 3 are 6 and minus 6 alpha. Now there is a problem here. See guys, if angular momentum has to be conserved, every term has to be 0. So if I take this guy, then alpha will be negative half. If I take this guy, then alpha will be minus 1. So either minus half or minus 1. Now the most appropriate option is minus 1, but this should not have happened. This question will actually become a bonus question because I can't get two values of alpha, because if I put minus 1, then this is not 0. If I put this as minus half, then this is not 0. So ideally the question framed over here is wrong. So it will be obviously kind of a bonus question because you should have a unique value of answer. So minus 1, you will mark it at the time being, but let me tell you guys, the NTA has been very, very evil this year. I mean, in JEE also there were many shifts, many questions which were deserving bonus, but NTA said we will not accept only bonus. Forget bonus, we'll not even drop the question, we will not even correct the answer, we'll not even correct the question. So I'm telling you, don't depend much on the bonus questions. If it is wrong, right, just take it for granted that you will lose the marks anyways if you get it. Okay, so it's very dicey and it was very unfair what NTA did on JEE students. I hope it does not happen with the NEET students. And the worst part is they charge 200 rupees per question for you to reevaluate, to reconsider that particular question to be dropped or for bonus. So just imagine the amount of money involved out there. It's crazy. Moving on, assertion. Diamonds are known for their spectacular brilliance, but diamonds found in nature rarely exhibit brilliance. By cutting the diamond suitably, multiple total internal reflections can be made to occur. What do you think about this? Okay. Sir, it's 12 minus 12. Did we do 12 minus 12 alpha? What did we do something wrong? Oh, sorry. Yeah, you're right. By mistake, I did not. Sorry, guys. This over here, yes, sorry. Then it is not a bonus. My bad. 2 6 are 12 and 12 minus 12 alpha. So it will be 12 plus 12 alpha. Okay, now it is fine. This will give me alpha is equal to minus 1. So then I should not give bonus. But even if it was bonus, forget it. Okay, cool. Yeah. So I think I made a small mistake over here while doing that. Anyways, coming back, what do you think about this question, guys? Is diamond related? When you cut the diamond suitably, multiple internal reflections happen inside a diamond. If this is a diamond, if multiple internal reflections happen, then what happens? You get a lot of shining, the twinkling of the diamond that you get. Yes, and diamonds are known for their spectral brilliance, but diamonds found in nature rarely exhibit brilliance. Why? Because only when you cut it in specific directions, then only you will get total internal reflection. So naturally occurring diamonds are not going to show that brilliance because they are not yet being cut. So yes, it is the correct explanation, and you need the cutting of diamonds by a proper person. Yeah. Very good. Excellent. Shall we move ahead to the next question? I think this question just got repeated again. My bad. Okay. So moving on. Let's see. The equation of the wave is given by blah blah blah. If x and y are measured in centimeters, the velocity of the wave. The velocity of the wave is always the coefficient of the term containing time upon the coefficient of the term containing x. So coefficient of the term containing time is basically the number 8 and coefficient of x is basically 1 by 8. So 1 by 8 and 8 goes multiplies and it will become 64. But remember, whenever there is a negative sign, then the wave is going in the positive axis. If it is a positive sign, it is going in the negative axis. It's exactly opposite. So 64 along the positive direction of x. 64 along the positive direction of x. Is that right, everyone? Very good. Perfecto. Let's go to the next question on your screen. To velocities of sound at the same pressure, two monoatomic gases, rho 1, rho 2, V1, V2, rho 1 by rho 2 is 4, then the value of V1 by V2 is. Okay, think about it. What is the speed of sound formula? Speed of sound formula. Densities, speeds, okay. Speed of sound is nothing but gamma P by rho. Same pressure is given. Monoatomic gases are also given. So gamma and P are going to be the same because they have clearly mentioned monoatomic gases and same pressure. So gamma is also same. So if I want V1 by V2, it will be rho 2 by rho 1, but rho 1 by rho 2 is 4, so rho 2 by rho 1 is 1 by 4, so it will be 1 by 2. So 1 by 2 means option B is the right answer. Perfect. Direct question on the speed of sound. Yeah, very good guys, proud of all of you. Let's go to the last few questions coming up on your screen. Displacement graph is shown, the force graph will be how? This is displacement graph. As you can see, y is nothing but a sine omega t. If y is like this, how will the force be? It will be minus ky. So it will be minus k a sine omega t. So it will be the sine curve, but it will be starting with the negative side first. So negative will be first, then positive side. So C option is the right answer. Yeah. So it is negative sign. That's all you need to consider. It is sine but flipped. That's all. Is that correct, everyone? Very good. Let's move. Let's move to the next question. The ratio of the speed of sound in nitrogen gas to that of the helium gas at this much temperature. Ratio of speed of sound. Now, speed of sound is root of gamma R T by M. Another formula if you don't want to write in pressure and density. Okay. Mass will be different. Gamma will be different. R and T are the same. So, speed of sound in nitrogen upon speed of sound in helium will be, it's proportional to root of gamma. So, gamma of nitrogen by gamma of helium, and here molecular mass of helium by molecular mass of nitrogen. Okay. Now, gamma of nitrogen, Cp by Cv value for nitrogen. Nitrogen is a diatomic gas, so it is nothing but 7 by 5. For helium, it's a monoatomic gas, so it will be 5 by 3. Okay, for helium, mass number 4, nitrogen 28. Okay, perfect. So 5 comes here, 7 goes on the top, 7 multiplied by 3 goes over there. 5 and 5 are there over here. Let's take a common root, and 4 by 28 is 1 by 7. 7 cancels. So it will become root of 3 by 25, which is root of 3 by 5. So root of 3 by 5, which is C option. There you go. That is the correct answer. Is that correct, everyone? Yeah, directly. Many people either forget gamma or they forget M. They then later on realize, oh, gamma was different. Oh, mass was different. So one of the things you make mistake, you will get some other option or none of these only. Cool. So moving on. The SHM equation is given by this. The equation A is the acceleration, X is displacement, the time period. Okay. Acceleration will be, take it on the other side, minus 16 pi square x. And we know acceleration is minus omega square x. Therefore, omega square will be 16 pi square. Therefore, taking roots on both sides, it will be 4 pi. But omega is nothing but 2 pi by t. Therefore, 2 pi by t will be 4 pi, by 2 and 4 goes cancelled. So t will be half a second, which is 0.5 seconds. So 0.5 is B option. Yeah, there you go. That is the correct answer. Isn't it? Very good. Shall we go ahead to the next question, my dear students? Here it comes on your screen. Okay, there we go. Magnetic field. After this, Diksha Mam will be starting. Magnetic field in a plane electromagnetic wave, blah, blah, blah. Question is, what is the wavelength? Wavelength. Just look at this coefficient of X guy. So whatever is the coefficient of X, so this was 6.28, and this power of 10 to the power 2 is also there, and K is nothing but 2 pi by lambda. So this is 6.28 into 10 to the power 2. Now we can directly cancel 2 pi and this because they are exactly the same. So 1 by lambda is 10 to the power 2. So lambda will be 10 to the power minus 2 meters, which is 1 centimeter. So 1 centimeter means option A. Yes, that is the correct answer. Okay, cool. So time to call upon Diksha ma'am. Okay, chemistry is starting. I'm ending the stream.
Hi students, welcome to Vedantu NEET English. I'm Diksha Kaushal, your chemistry master teacher. And everyone, let us start with the chemistry part. So you have done bio, that is botany, zoology and physics till now. And now let's start the questions of chemistry. Okay, so everybody, shall I start with the class? Everybody, I want to have green hearts and fire in the chat box. Because I know it's a long time you are studying, ma'am, 10 a.m., ma'am, we are studying. But it's okay. Why? Because these questions are the questions that are going to get repeated in your exam. You can see in 2024 as well, the topics, the questions that we took in the mock test, right? Same question or the question surrounding the question that we have taken in the mock test were there in the exams, right? Exact, some question were exact copy paste. So that's why I'm saying take these mock tests very, very seriously, okay? Now let us start the questions without wasting any time. And I want all of you try to answer the questions by yourself as well, right? I don't want to see any chit chatting in the chat box. Rather than I want to see your answers in the chat box. Okay? Yes. Now let us start with the very first question on your screen. That is, there is no SS bond in the following, right? Okay, they're asking about there is no SS bond in the following. Yes, in the following, so you have to know the structure of all of these. Ma'am, how you will get to know the structure of all of these? These are in P block, and in P block, ma'am, but in P block, structures are not there in our exam. So I would really, really advise you to do the structures of oxo acids, okay? Oxo acids are very important, especially of phosphorus and sulfur. From here, directly they can ask you the question which can be answered in just seconds, right? So here it is. S2O7 has no SS linkage, as you can see here. This must be your H, this must be your H. So here it is having SOS linkage, not SS direct linkage, right? So that is why answer is going to be S2O7, that is D is the correct answer. This is a direct fact-based question, right? So for these fact-based questions, side by side in these questions, I'll also tell you that to answer these kind of questions, what are the topics that you need to do. For example, here I have mentioned that P block oxo acids of phosphorus and sulfur mentioned in your NCRT. Mentioned in your NCRT that you have to do for sure, right? You don't need to do the reactions of P block, but you should do the structures of P block that are very important for you, right? Next is, which of the following compound has less thermal stability? Okay, less thermal stability. If you talk about the thermal stability, right? So here, as we go down the group, right? As we go down the group, what you will see, size will increase, right? Size will increase. That is why if you see down the group here, size is increasing. So if you see on that size, the charge is same, right? So that is the reason when size of this cation, right? They're asking you about the size of the cation. When you talk about the thermal stability or simple stability, that is directly proportional to the size of the cation. Size of the cation. So as you go down the group, the size of the cation will increase. That is why the thermal stability will also increase here, and hence LiNO3 will have least thermal stability. Okay, it will have the least thermal stability. That is your E is the correct answer. Okay, E is going to be the correct answer here. These are the kind of questions that students usually skip but these are the rank deciding questions, okay? Right? These are the rank deciders, right? So let us see the next question here. The relationship between osmotic pressure. At 273 Kelvin, when 10 gram of glucose, 10 gram of urea, 10 gram of sucrose are dissolved in 250 milliliter of water. Now, if you see this here, we know that pi is equals to, that is osmotic pressure is equals to CRT. RT, we know that it is constant. C, concentration means molarity. That is number of moles. What is number of moles? Weight by molar mass, right? This is volume, this is in liters, and this is RT. If you see this here, the gram are same, the mass is same here. That is why we can say pi is inversely proportional to the molar mass of the solute. Okay. So more the molar mass of the solute, less will be your osmotic pressure. According to that, you have to answer this. All of them are non-electrolytes, so there is no Van Hoff factor that is involved here, right? So directly osmotic pressure is directly proportional to number of moles that given in the non-electrolyte. Hence, C is going to be the correct answer. That is, third is what? Third is sucrose, then you have glucose, then you have urea, right? Okay, so that is going to be the correct answer for your, according to the molar mass, right? Okay, according to the molar mass, you can decide here. Sucrose you have studied already in biomolecules, right? This also you have studied in biomolecules, right? Urea, we know that is, urea is CH2NH-CH2, okay? That is your urea, right? So that is how like things will become very easy for you, right? Next we have is among the following least stable resonance. Achha, least stable. Now, if you talk about the resonating structures, there are few points that you need to remember. Okay, one by one, I am writing these points for you, right? Okay. First is neutral is more stable as compared to cation. Any charged. So, firstly, you have to see if there is any neutral species here. Ma'am, none of them is neutral. So, answer will be first, we cannot apply here. Next is you have to check the pi bonds here. Now, everybody check the pi bonds here. How many which has more pi bonds will have more stability? It has 2 pi bond. This also has 2 pi bond. This also has 2 pi bond. From there also we can't identify which is more stable, right? Then remember that negative charge is stable, more stable down the group and across the period. Down the group, along the period, negative charge stability is going to increase, right? So that is why what will happen here, if you see negative charges on oxygen, negative charges on oxygen, okay? Negative charges on oxygen, negative charges on carbon, carbon, and this is also carbon. That means here also clearly we can't identify anything like specific here, right? So this point is also not valid here. Last point is charge separation. Last point is charge separation. So students, charge separation is inversely proportional to stability. More the charge separation, less will be the stability, right? If you see here, the charge separation is same. This thing is same. There is no change over here. If you see, the change is wait a minute. This thing is already eliminated. Can you know why this is already eliminated? Because if you see, this is positive, negative. This is positive, negative. Here you have only negative. So this one is already eliminated here, right? Okay. So if you see among these two, there is no as such thing regarding charge separation. This is positive. This is negative. This is negative. This is positive, right? So when you have same charges, that is unstable. When you have opposite charges on the adjacent position, that is going to be more stable. That means the last point is also not valid. So what we will say that when you have, let's say you have positive, positive, due to similar charges on the adjacent atom, structure is least stable here. Understood? All these points are not valid here. This is not valid. This is not valid. This is not valid. This is not valid. In the end, we got a conclusion. They are asking you the least stable among them. least stable among them, right? Now, if you see, this is same, this is same, this is same. This part is different here. Here you have positive, negative. Here you have only negative. Here you have negative, positive. There is charge separation here, right? And here, what you will observe, this is positive, negative. But when you have two same charges on the adjacent position, right, because of the repulsions, it makes it very unstable. So answer will be least stable. Is A is the least stable. Okay, that's how, why I mentioned all the points as well, because this is one kind of question. Okay, they can even ask you any question related to one of the points here, right? So that automatically becomes very important for you. Next, students, we have which one is most reactive towards nucleophilic addition reaction. Okay, a nucleophilic addition reaction, that means nucleophilic, someone who loves carbon with deficiency. We need to see where carbon is very poor, very electron deficient. Nucleophile will attack very easily over there, right? So here you can see no effect. Here we know that already it is donating the electrons. Here if you see, this is CHO, right? And here it is withdrawing the electrons, right? Okay. Here also to this CO also it is donating the electron from this side, right? And here what is happening, right? This pi bond will shift here, this pi bond will shift here, this also is donating. So this carbon is also, this carbon is taking electrons from resonance and inductive. This C double bond is taking electrons from inductive, though the distance is very large, right? Here you see no effect. But in this case, you see a electron withdrawing group. Suppose you have like 1,000 rupees. Someone takes out all the money from you. So it's like benzene has a lot of money. Benzaldehyde a lot of money. And NO2 is taking out money, making the carbon electron deficient, right? Okay. So that is why if you see the maximum they were asking, no, most reactive is going to be, this is going to be your most reactive here, because this is very electron deficient, right? Okay. This is very least electroreactive because it is getting from two positions, right? One is plus side. Next is, you see here, from mesomeric effect also it is donating the electron. Here it is a weak effect, that is giving the electron, that too from the distance here, right? So that is why this is going to be the reactivity order here, right? Okay, students? Yes. Next is, now this question, right? Which of the following facts regarding ionic conductance of the following at room temperature is correct? Okay. These kind of questions have a very high probability that they can ask you in the exam. Again, I am saying because these are the kind of questions that they have asked in JEE April attempt, right? Okay, so we can get a mirror from there. So what are the questions that they are asking from? NTA is the same agency that is conducting the two exams, right? So this is the question that has been repeated 2 to 3 times in the April attempt. Which of the following facts regarding ionic conductance? So one thing about ionic conductance you should know, that ionic conductance is directly proportional to, students, ionic conductance is directly proportional to what? You can just write here. It is inversely proportional to the size of ion. Or you can say size of hydrated ion. Size of hydrated ion. See, smaller is the ion, larger will be, more the number of water molecule will get attached to it. More water molecule get attached to it, that means the size of the hydrated ion of that ion is going to be larger. That is why that is the size of ion. Okay. So more is the size of ion, more is the ionic conductance here. So if you see this here, yes, answer is going to be, this is going to have more size of ion. Down the group, size of ion increases. Down the group, the size of hydrated ion decreases, right? That is why this is going to be the order, right? Okay. Just remember this particular thing here. Just remember this particular thing here. From here, very interesting questions they can ask you, right? Next is, which of the following is paramagnetic? Now, this is a standard question, okay? This will be, it's like every exam, right, some questions are standard questions that will be there in your exam, right? You will see these kind of questions in every mock test. Why? Because I want you to go through all the questions that are there from these important topics, right? Okay. So which of the following is paramagnetic? So students, if you see this here, paramagnetic here, O2 2 negative, see, O2 is paramagnetic. When you add 2 electrons here, it is going to become diamagnetic, right? N2, let us discuss C2, NO2. Chalo, let us discuss one by one. So firstly, I would advise you to discuss N2 here, right? N2 is 14 electrons. 14 minus 8 electrons in KK star, okay? 8 electrons in KK star. If you see this here, it is going to give you how many electrons? 14 minus 8 is 6 electrons it is going to give you, right? Now here, what you will have, 2, 1, 2, 1, that is why, because it is less than 14. So answer will be 1, 2, right? 3, 4 and 5, 6, right? So here that is why it is going to be diamagnetic, right? So that is your diamagnetic species, diamagnetic, right? Now let us talk about N2 positive. See, if it is diamagnetic, N2 positive is going to be paramagnetic. Let me tell you why. Because one electron it will take out. See, simple thing is, if you apply the logic, you don't need to see C2, O2, 2 negative. This is not even needed. Because logic says that, wait a minute. When you take out one electron from here, it is going to have one unpaired electron. That means it is going to be paramagnetic in nature, right? Okay, one electron you will take out from N2 only, right? So that is why answer will be C is the paramagnetic species that you will have. Rest of them are diamagnetic in nature. Okay, rest of them are diamagnetic in nature, right? Understood? Yes, students, right? Now, next one is the Bohr orbit radius for hydrogen. Again, standard question. Very, very important question. Mark my words. One question will be there based on Bohr model, right? The Bohr orbit radius of hydrogen atom is approximately this, the radius of first excited state. That is, here they have given N2 is equals to N is equals to 2 also. But in the exam, the chances are they will not give you N. They will give you, it belongs to first excited state. That means N is equals to 1 plus 1. That is, second excited state means 1 plus 2. Third excited state means 3 plus 1. Fourth excited state means 4 plus 1. Okay, so that is how you are going to see your value of orbit. So directly here we will apply the formula. R is equals to 0.53, that is the radius of ground state, N square by Z. Z is your hydrogen atom. So for hydrogen atom, Z is equals to 1 only, right? Okay. This will be Z is equals to 1, right? So here N value is 2. You'll just put this, see the answer there in Armstrong. So you don't need to convert it to anything else. Otherwise, the conversions, Armstrong to nanometer, picometer, all these conversions are given in your some basic concepts of chemistry, first chapter of chemistry that you have studied in class 11. Go through that conversion. Conversions are very important for your NEET exam, very important. All the conversions, joule to electron volt, then the conversion of joule, kilojoule conversion is very simple for you. Then the conversion of Armstrong, picometer, nanometer, all these conversions are very, very important for you, right? Next, wait a minute. I think the question I forgot by mistake. One slide is being deleted from here. Okay, I know the answer is here, but let's see the question. On the electrolysis of aqueous solution of sodium sulfate. Now, students, this is from product of electrolysis. Product of electrolysis. Ma'am, now you know what the question. Ma'am, is this important? Yes, this is important, right? This is not a very, this is not a very that kind of topic that every student knows. Trust me on that. Most of you will be blank here, right? Ma'am, I don't remember that. Right? That's the reason I'm saying, is this important? They are asking these kind of questions, NT's other exams like JEE mains, In April attempt, they have asked around 2 to 3 questions based on product of electrolysis. That means that automatically becomes important for you, right? So that is a new trend. Okay, so some questions are standard questions. Remember one thing, understand, some questions are standard questions. Some questions, definitely they will give you some new kind of questions, and these are the new kind of questions that are not so repeated. You will not very frequently see product of electrolysis in your previous year questions, right? But 10 years down the line, this will be the important topic. So that is why the initiation is from your exam, right? So go through the product of electrolysis in my class 12th marathon that we took, all the teachers, we took it on last Thursday, right? So go through that. In that electrochemistry, I have given you one table where I have just directly given you at anode and at cathode which will be formed. Okay, go through that table, right? So now on the aqueous solution of sodium sulfate. So if you see sodium sulfate, this Na positive and SO4, 2 negative is also there. But exactly what will happen? At cathode, you will have H2. At anode, you will have the formation of O2 because H2, water, has more reduction potential as compared to sodium, and water has more oxidation potential as compared to sulfate. That is the reason, very important, H2 and O2 is formed here. So answer is going to be, they are asking you at cathode, you have the formation of H2 here. You can get confused between sodium and H2 here easily, right? Okay. Next question is cryolite, right? Okay, cryolite is dash is used in electrolysis of alumina for decreasing electrical conductivity. This used in electrolysis of alumina for lowering the melting point of alumina. This, okay, they have given you formula is right only in all the cases, so we can't differentiate it based on formulas. Used in electrolytic purification, used in oxidation. Now you have to tell me the uses here. Electrolysis of alumina, used in electrolysis of alumina for lowering the melting point, purification of this, what it is related to. So basically, it is used to lower the melting point. Okay, used in electrolysis of alumina for lowering the melting point here. These are application-based questions. Okay, application-based, fact-based questions. Basically, they are, right. Why factual-based questions? Yes, that is also the new trend, that fact-based questions they are asking you, right? Not too many fact-based questions, but yes, you can see in your paper of chemistry around 2 to 3 questions from inorganic chemistry that are factual questions. Okay. Now, next is which statement is correct. Okay, so 4 statements are given to you. Again, a standard question. Okay, this is not a new question. This is a standard question. After 20 years also you will see VBT, Vesper, MOT in the exam of NEET and JEE. That will be there, right? Very important these are, is correct. So you have to know that which of the following is basically, first is tetrahedral, then there is which of the following is paramagnetic and diamagnetic. You need to see which of the following is the correct answer here, right? Okay. Yes, everybody. So one by one we will see, right? Okay. Square planar. First is, let us eliminate. This is sp3, this is sp3, this is sp3, this is dsp2. Okay. Firstly, I'll just see whether NiCl4 is dsp2 or not, right? So if I see NiCl4, X is going to be, this is X minus 4 is equals to minus 2, nickel is in plus 2 oxidation state, means 2 electron from 4s will be eliminated. You will have 3d8 here. It is 4s to 3d8, right? So 2 electrons will be eliminated. You will have 3d8 here. Now, here we know that this is a poor, like it's a weak field ligand. So what will happen here? 1, 2, 3, 4, 5. This is S, this is P3. Now, here you have 8 electrons. So there is poor fielding. So 1, 2, 3, 4, 5, 6, 7, 8. No pairing will be there. It is paramagnetic, yes. It is sp3. So that is automatically a wrong statement here. Now let us see for NiCl. So here it is X is equals to, again, positive only, but because of Cn, it will cause pairing. So in the same thing, what will happen? There will be pairing. Okay, in the same thing, there will be pairing. So what you will see here is 1, 2, 3, 4, 5, 6, 7, 8. So it is DsP2, right? Here it is given to you, that is tetrahedral, that is sp3. So this is again eliminated, right? Now you have NiCO for you, right? Third one is NiCO. Now, in this case, this is a neutral ligand, right? That is why nickel exists in zero oxidation state. Nickel in zero oxidation state is 4s2 3d8, right? Total it has 10 electrons, right? Okay. Now here what you will do, again you will draw that. Now tell me carbonyl, right? That is CO, right? It will cause pairing or not. It is a strong field ligand when it comes to Ni2 positive. So it will cause pairing. So it will be total 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This is sp3 and this is diamagnetic in nature. This is sp3. This is diamagnetic in nature. So answer will be C is the correct answer. Right? Okay. So that is how you're going to solve the questions of VBT from coordination chemistry. Very, very important. Right? Next question is the products formed in the reaction of cumene with O2 followed by treatment with dilute HCl. Now this I expect you can answer it in within seconds, right? So you have cumene. Now ma'am, what is this cumene? Cumene is when on benzene you have iso. This is CH3, this is also CH3. Isopropyl benzene is cumene, right? So now the product formed in the reaction of cumene with O2 followed by treatment with dilute HCl here, right? That means this is the cumene process where you will have here peroxide linkage, that is OOH, and this is CH3. And after this you will have the formation of acetone and phenol here. So answer will be B is the correct answer. Very good. Phenol and acetone is the correct answer, right? Next question is dissolving 120 gram of urea. How much urea you have? 120 gram, right? Yes, the answer is definitely right, guys. The answer is right. Dissolving 120 gram of urea in 1000 gram of water gave a solution of density this, the molarity of the solution. Now, what is molarity? Molarity, if you discuss, that is number of moles. Ma'am, what is number of moles? Number of moles is weight divided by molar mass, right? Molarity is volume of solution in, let's say, we are giving it in milliliters. Why? Because this density is also in milliliters, right? So, to ease this, I'll just consider this in milliliters. Now, if you see dissolving this much of urea, that means W2 is given to you. Molar mass is also given to you. 1000 gram of water. We know that density is equals to mass by volume. So, volume is equals to mass by density. Mass is given to be 1000, right? And density is given to be 1.15, right? So, this is in water. That means we are talking about solution here, right? So, this is the, wait a minute. In 1000 gram of water gave a solution of density. Mass is given to us of water, right? We are calculating the volume of solution, right? So that means mass we will take it of solutions only, right? Solution is what? This is 120 gram, this is 1000 gram. So it will be 1120. Mass is going to be 1120. Density is going to be 1.15. Solving this, you will have your answer of volume in milliliter. You put it here. Answer will be molarity is equals to, this is 120, right? This is divided by molar mass is 60. This is 1000. This is density is 1120 and this is 1.15. Solving this, you will get your answer here. Okay. Solving this, you will get your answer of molarity here. Right, B you are saying, okay, very good. B is the correct answer. Very good, students. Right, so answer is, C is coming out to be the correct answer, yeah. C is coming out to be the correct answer. B you are saying, let me just see, 2.05. So it is coming out to be 2.05. How many of you actually calculated it, right? So it is marked here 2.05, but let's see if your answer is coming out to be 3 point something, then that can also be the correct answer, right? Okay, so just calculation thing, right? Okay, I think C should be the correct answer, right? Okay, next question is a metallic carbide. Now this is a very interesting question. Okay, a metallic carbide on treatment with water gives colorless gas which burns readily in air, which gives a precipitate with ammonical silver nitrate solution. Gas evolved is what, right? So they are telling you there is some metallic carbide. So if you remember, there is CaC2, metallic carbide. On treatment with water gives a colorless gas, right? It will give you which burns readily in air and which gives precipitate with ammonical silver nitrate, right? So with ammonical silver nitrate, it will give you ethine. This kind of situation. They're asking you the gas involved is. So if you see this here, CaC2 in water, it will give you C2H2, that is ethine, right? This is the gas that is evolved here. Now this particular gas with ammonical solution, we know that this will convert this hydrogen and this hydrogen to Ag. So it will give you a tolerance test. We say it as tolerance test, though it does not give silver mirror. It gives you white precipitate with silver. With copper, it will give you copperish color, right? Brownish, reddish color, right? So it is giving you white precipitate here. So this gas was what? This gas was nothing but this is called acetylene, right? Okay, understood, students? Next is for a reaction H2 plus I2 give rise to 2HA, the standard Gibbs free enthalpy is greater than 0. The equilibrium constant would be. Now you should know the formula here, that the formula between delta G and the formula of K equilibrium, right? So this is the formula, delta G is equals to, I'll just write it here, delta G naught is equals to minus nRT natural log of K. Simple thing is they are giving that this is positive. If this is positive, this must be negative, right? That means ln of K should be negative, right? Ln of K is only negative if K is less than 1. So that means the answer is K should be less than 1. Log of anything is negative when that thing is less than 1. Log of anything is positive if that thing is greater than 1, right? Okay, next question on your screen is, which of the following statement is not correct? Which of the following statement you see is not correct, right? Okay. Now, right? Is it Varsha? Don't worry, I'll see that, Varsha. If that is there, if you have 100 plus comments, then I'll surely take that today itself or tomorrow, right? Okay, now which of the following statement is not correct here, right? LaOH thrice is less stable than this. In lanthanide series, ionic radius of this decreases. That is true. Lanthanum is actually an element of transition rather than lanthanoid series. That is also true. It belongs to group number 3. Lanthanum belongs to group number 3. After lanthanum, you have lanthanoid series, which belongs to F block, right? Atomic radii of zirconium and hafnium are same because of lanthanoid contraction. That is also true. So La, that means the hydroxide of lanthanum is less basic than lutetium. That is the wrong statement here, right? So as you go down the group, the basicity will decrease here, right? That is why larger in size than this, so it has more polarizing power, more ionic, that makes it more basic, right? So basicity will decrease down the along the lanthanoid series, and that is the reason lanthanum hydroxide is more basic as compared to lutetium, right? Next is, in ethane and cyclohexane, which of the following pair conformers are more stable? Now, if you see the conformers of ethane, you have staggered and you have eclipsed, right? Eclipsed is like this, eclipsed is like this, which has more repulsion. But staggered is like what? Staggered is like this, okay, which has less repulsion, right? So obviously answer will be staggered here, right? What about Cyclohexane. What about cyclohexane? In cyclohexane, it is chair conformer that is the most stable here. So answer will be B is the correct answer, staggered. And then you have chair conformer for cyclohexane, which is most stable. This also I have taught you in class 11th, like full chemistry where we did, I have taught you that there, right? How many hyperconjugating how many hyperconjugable hydrogen atom does this molecule have? So what you need to see is you need to see the alpha hydrogens here, right? How many alpha hydrogens you have here? If you see, this is alpha, this is alpha, this is alpha carbon, this is alpha carbon. This has 3, this has 2, this also has 2. So it will be 3 plus 3, 6, 7, 8, 9, 10. So total you have 10 hyperconjugating structures. So answer is going to be C is the correct answer. Very good students. Next is 2-bromopentane. Now what you have is 2-bromopentane is heated with potassium ethoxide, right? In ethanol, the major product obtained is, what is potassium ethoxide, right? This thing. Now, firstly, you tell me what exactly is the thing that they are asking you. What exactly is the reaction? What will happen here? Let me see. Because here to identify that is not very difficult, but if you identify that would be great, right? So you have 1, 2, 3, 4, 5, right? On 2 you have bromo, right? So when you have any halide and you have a base, right, that is acting upon it, right? So what happens? What is this base, ma'am? This is basically QH in alcoholic medium. That is how it has become C2H5OK, right? So in alcohol, when you add base, it will become C2H5OK. So directly they have given you that. That means this is dehydrohalogenation reaction, is it? So this will be eliminated. Now hydrogen will be eliminated from this side. Either it can be 1, 2, 3, 4, 5, this answer, or it can be this answer. Because hydrogen can be eliminated from this side or hydrogen can be eliminated from this side. But we want ZZ product, that is more alkylated product. So answer will be, this is the answer that you will get here. That is pent 2E, right? Okay, so that is what you will get here, right? Ethoxy is not given to us. If you see trans or cis, you will get here. So you need less repulsions here, right? So for less repulsions, you will get here, wait a minute, yes. Trans isomer is formed because of less repulsions here, right? Okay, so answer will be trans pent 2E. Because see, that product is more stable, which is less, like less repulsions are there. If you see, talk about trans, this is trans and this is cis. Cis definitely has more repulsions as compared to trans, so that is why trans is preferred over cis, right? Next is, which of the following reaction can produce RCOAR? Okay, this is basically R, this is C double bond O, and this is benzene ring. Okay, if you see this here, you have benzene in which you are adding, this is, it will form, this is benzene, and here you are adding C double bond O and again one more benzene, because this is the electrophile that will be formed here, right? Here you will have this electrophile, but here you have RMGX, and this is acid hydrolysis, right? So definitely you are not going to have, because what will happen here, no? So here what will happen? This is RCO, right? So this is R, this is C double bond O, right? So what will happen here? This is Cl. This ARMG will attack here and this will be converted to OH here, right? And hence you won't have, this is acidic hydrolysis. After this, wait a minute, you will have OH. Then this OH will be eliminated, this H will be eliminated. Wait a minute. Okay. Wait a minute. Last one is, you have RCOCl. This is RCO will be formed here. This is Friedel-Crafts acylation. This is benzene. And this is COR. Right? Directly that thing will be formed here. Right? So they are asking you ArCOR. So maximum chances of the following reaction can produce RCO. That is this D, that is Friedel-Crafts acylation. Alkylation will produce you this. On benzene, you have one COR product, right? Okay, so that can produce this Friedel-Crafts acylation reaction. Which of the following ion is colored? Now you have to tell which of the following ion is colored. Cu positive, Cu2 positive, titanium 4 positive, vanadium 5 positive, right? So Cu2 positive or Cu, right? So out of this, where you find a blue color solution of copper, right? So if you see this, if you don't want to say this directly, what you can see the electronic configuration here. This has 3D10, so it will be colorless. This has 3D9, so it will be colored, right? This has titanium is 22, so it will be 4s2, 3D2. You take out 4 electrons, it will be 3D0, which is colorless. Vanadium is 23, so it will be 4s2, 3D3. You take out 5 electrons, it will be again 3D0, which is again colorless. So answer will be B is the correct answer here. Next we have is a molecule of AB undergoes trimerization in benzene. Its bond Hoffe factor is 0.6. The degree of association of AB is going to be what? Right? Degree of association they are asking you. See, directly you will apply this formula here. See, either you can solve it like this or you can directly apply the formula here. Right? Totally your choice. So that is I is equals to 1 plus alpha 1 minus 1 by n. Right? Okay? I think here also it should be minus. Yes. Okay. Now, if you put the value of n, it is trimerized. So answer will be I is equals to 1 minus alpha. Right? This is going to be 1 minus alpha. This is going to be minus alpha. This is minus alpha by n. So that is 1 minus alpha by 3. Right? So that this is what given to you here. It will be positive. Okay? So either directly you can put the value according to dissociation or association. Right? Now association is given to be 60%. Right? 0.6. The value of alpha is given to be 0.6. So directly you will put the value of alpha, that is 0.6 here, and you will get your answer here directly, right? Or what you can do, I would advise you directly go for formula, no need to do this, right? Here we know that, okay, this is being dissociated. For general formula, if you put n here, this will be n. This is the formula that you will get, right? But if you see, ma'am, if I don't remember the formula, you can take initial as 1, right? Final as 0. Then 1 minus alpha will be taken out. This will be alpha by 3, right? And after that, number of moles at equilibrium is add all these moles divided by initial was 1. So you take this, you will get your answer here. But I would again advise you to go through, just directly use the formula. Trimerization n is equals to 3. Dimerization n is equals to 2. Directly put the formula and you will get your answer here. Right? Okay. Next question is, enthalpy of reaction is given to you is negative. If enthalpy of combustion of CH4 and CH3 are X and Y respectively, so now they are asking you enthalpy of reaction is given to you which is negative, that means it is exothermic in nature. If enthalpy of combustion, that is CH4 and of CH3OH are X and Y respectively, select the correct option here. Right? Okay. So, if you say overall delta H should be negative, so that is delta H of product minus delta H of reactant should be negative, right? For that, delta H of reactant should be greater than product, isn't it? So here, that means delta H of reactant should be greater than product. That means CH4 should be greater than C2H5. So we can say X must be larger than Y. That means X is for this. Okay. Reactant means you have CH4. Product is CH3OH. So this is X and this is Y. So definitely X should be greater than Y here to have this value. So answer is x is greater than y. Okay, understood? Everybody, right? How we got this answer? Exothermic, negative. From there you got the hint that delta H is negative. According to that, you can solve this, right? Next is, now again, very important question. Arrange the following oxidizing agent in order of their strength under standard state. See, first thing is always remember reduction potential is directly proportional to oxidizing. No. More the reduction potential, right? More is the power to get reduced, right? Okay, to oxidize something is more, right? That means if I have more reduction potential, I myself want to get reduced, right? I want to oxidize something, so my oxidizing power is going to be very high, right? So oxidizing power is very high, right? So you just need to see which has more reduction potential. First, you have to see are all of these reduction potentials only, right? This is 0 and this is -1. That means oxidation number is decreasing here. For reduction potential, oxidation number should decrease only. So this is reduction potential. This is also oxidation number is decreasing. Here you have +6 and this is +3. How you calculated +6? 2x minus 14 is equals to -2, right? 2x is equals to 12, x is equals to 6. So that means here it is +6. So +6 to +3, again this is reduction potential, right? Now what you will see, if all of these are the reduction potentials here, right? This has the maximum reduction potential, this has the maximum oxidizing power. So maximum will be of CrO7. Then you will have Br, right? Then you will have Fe3 positive, that is E is going to be the correct answer. Students, please don't directly blindly see that, okay, this must be reduction potential. You have to check whether all of these are reduction potentials or not. Might be they will give you some oxidation potential. You have to put a negative sign, convert it into reduction potential, then see which value is larger or not. Got it? Yes, everybody? Yes, students? Okay, let's make the likes by 1000 plus, okay, by the end of the session. Let's make the likes by 1000 plus by the end of the session. Which is the correct order of increasing energy listed orbitals in the atom of titanium? Doesn't matter it is titanium or what, always apply N plus L rule, right? N plus L rule, N plus L rule. If you see, this is for 3s, then you have 3p, then you have 4s, then you have 3d, right? So here I'll just write it here. If this is 3s, 3s means 3 plus 0, that is 3. This means 3 plus 1, that is 4. This means 4 plus 0, that is 4. And this means 3 plus 2, that is 5. Okay. And between these two, more the value of N, more will be the energy. So that is why 3p has less energy as compared to 4s, right? So answer will be, increasing order is, first you have 3s, then you have 3p, right? Then you have 4s, then you have 3d. This was for those students who have no idea about the energy orbital diagram. If you, not energy orbital, do you have no idea about the increasing order of energy? If you already know the increasing order of energy, that is on your tip, so you don't even need to do that. So this is 3s, 3p, 4s, 3d. So answer is D is the correct answer here. Right? I can see you are typing D. In which of the following complexes a central metal atom is D2sp3 hybridized? The complex is diamagnetic in nature. Okay? The complex is diamagnetic in nature here. Now students, you have to see all of these, right? If you see, this is cobalt 3 positive. For cobalt 3 positive, diamagnetic they are asking you in D2sp3, right? Okay. Oxalato, when you have cobalt 3 positive, it will behave as a strong field ligand. NH3 is a strong field ligand only, so it will cause the pairing. Here. Right? Okay. Now here, let us see what we are going to do. So if you see, this is cobalt is in +3 state. Here also cobalt is in, this is +3, this is -3. Again, cobalt is in +3 state. How? X minus 6 is equals to -3. Because this is auxiliary is bidentate ligand, right? So both of them are in +3 states, right? So cobalt, we know that that is 27. This has 4s2 3d7. You take out 3 electrons, it will be 3d6, right? That means here what you will have, here what will you will have, 3d60, both will cause pairing, so both will have the same diagram, okay? Both will have the same diagram. 3 6 means 1 2 3 4 5 6, so that is D2sp3. D2sp3 is going to be the correct answer here. But if you talk about Zn, in zinc, let us see, right? In zinc, let us see, this is NO3 minus 1 here. So this is, wait a minute, okay, this is outside, right? This is outside, okay? This is water, right? So this is -2, this is +2. That means zinc is in +2 oxidation state. Zinc, we know that that is 4s2 3d10. +2 oxidation state, that means it is going to lose 2 electrons here. So answer will be directly 3d10. Now this is a weak field ligand if you see. Even if you see this is a weak field, strong field, doesn't matter. Because this is 3d10. So always it is going to show you which kind of hybridization, guys. What kind of hybridization it is going to show you. Everybody, if you have 3d10, even if you have pairing or not, it is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. That will be sp3d2. Okay. So answer will be that only 1 and 2 are going to show you D2sp3. Right? Okay. So 1 and 2. And if you see, both are diamagnetic, or diamagnetic means no unpaired electron. Here you have no unpaired electron. So 1 and 2 are D2sp3 and there is no unpaired electron here. This important part here was that oxalato, when you have cobalt 3 positive, it behaves as strong field ligand, it will cause the pairing. This was the important part here. This was the important part of this, so you know that it will cause pairing. This was the important part, right? Okay. Which of the following order is incorrect? Incorrect order, right? So based on orders, 1 or 2 questions definitely is going to be in your exam. 1 or 2 questions, definitely, right? You will see from P block around 2 questions from orders, right? 1 question from D block. D block may or may not be from orders, because in D block there are so many other things as well. So in P block, the chances are they will ask from exceptional orders and acidic, basic, strength, all of these questions regarding oxides and hydroxides. And from D block, the chances are they will ask you from KMNO4, K2CR7, and lanthanoids, all these electronic configurations they can ask you, right? Incorrect order, if you see here, incorrect order, students. Oxidizing power, bond length, if you see down the group, it is increasing. Oxidizing power is right. Bond dissociation enthalpy, if you see. Bond dissociation enthalpy, if you see. See, if you talk about F2 here, F2 is it is very, very small. It is the smallest element in this halogen group, right? In the entire periodic table also, it is the smallest element. It has 3 lone pair of electrons. So if you place 2 F2 atoms side by side, which are very small, and those have 3 lone pairs, so they will definitely repel each other. Because of the repulsion, F2 will not be that much stable to have the maximum bond dissociation enthalpy, right? So answer is incorrect order is C is the incorrect order. F2 is not having the maximum. First you have Cl2, then you have Br2, then you have F2. Reason I have told you, because of the electronic repulsions of the lone pairs here, right? It is very small. Same happens in O2, right? Same happens in N2, but it does not happen in C2. In carbon, C2 molecule, it does not happen. Carbon has the maximum bond dissociation enthalpy in its group, but O2 does not have, N2 does not have, and F2 does not have, because they all have lone pairs and because of that it causes repulsions, right? Next is, which of the following compounds cannot reduce Tollens reagent? Tollens reagent, what you need is aromatic or aliphatic aldehydes you need, right? Formic acid is one of the exception, right? It does reduces Tollens reagent, right? Then you have, this is ketonic, this is alcohol, and this is ketonic part here, COCH2OH, okay? This is ketone, so this is not going to basically oxidize this compound. Ma'am, how come this is going to oxidize? This is CH3, C double bond O, CH2OH. So when you have on the alpha position you have alcohol, right? Okay, in that case basically alpha hydroxy ketones, hemiacetals, formic acid. These are the three exceptions, not exceptions. You can say, usually we just remember that aromatic aliphatic aldehyde we need for Tollens reagent. But here is where you will get confused. Ma'am, this is aromatic, this is aliphatic aldehyde, this will show. Formic acid is also most of the students know, but this is where students do mistake, right? Alpha hydroxy ketone is going to show you Tollens test. Hemiacetal is going to show you Tollens test here. So answer is C is the incorrect, C is the one that is not going to show you any Tollens test. Next is dry distillation of sodium propanoate with soda lime decarboxylation. Dry distillation of sodium propanoate, right? Propanoate is going to give you which thing? It is definitely going to give you alkane. And that alkane will be one carbon less here, right? So answer is, this is propanoate. So answer will be, students, here you will have 2 carbons, right? So basically what always happens, this is RCOONA. This is always going to give you RH. That is 1 carbon less alkane it is going to give you. That is your ethane is the correct answer. If they have used butanoate, then answer will be propane. Okay, so 1 carbon less should be there. But in Coles electrolysis, I'll just write it here because this also they can give you. Coles electrolysis, if you have RCOONA, you get RR as the alkene part. You get RR as the alkene part here, right? Next is, you have, which of the following oxides of chromium is amphoteric? Now based on amphoteric, like tin oxide, lead oxide, chromium oxide, vanadium oxide, aluminium oxide, zinc oxide, all these are amphoteric in nature. But in chromium, which of the following oxides is amphoteric in nature? Anybody? You have Cr2O3, which is basically your amphoteric in nature. Lower oxidation state makes the oxide basic. Higher oxidation state, it is acidic. Always remember, if you are given with only one, see, if they ask you directly, what are the oxides? Tin, lead, aluminium, zinc, and chromium and vanadium. These are the oxides that are amphoteric in nature. Now, if they ask you from only one metal is constant, always remember that acidic character is directly proportional to oxidizing number, oxidation number. Okay, understood? Or you can also write it as directly proportional to number of oxygen atoms divided by number of central metal atom. That is the trick here. Okay, so you can also write it like this. But in case of oxophosphorus acid, it does not follow the trend. In case of oxophosphorus acid, it is opposite. More is the number of oxygen, less is the acidic character. It is opposite in case of oxophosphorus because of the hydrogen, because of the structure, right? So here it is. You can see this is 3/2. This is 3, this is 1, this is 5, right? So here answer will be, if you see higher oxidation state, no, This is 1, this is 3, this is 3 by 2. So then, acha, right? They are asking you the acidic. Okay, so this is 1, this is 3, this is 3 by 2, that is 1.5, this is 5. Among this, if you see, this is in the middle. That is why this is amphoteric in nature, right? Okay, in the middle, that is why it is amphoteric in nature. So answer will be C is the correct answer. If they would have asked you acidic, then the answer would be this, which is having more oxidation number, is having the more acidic nature, right? Next we have is which is the minimum pH necessary to cause a precipitate of this to form this much of PbCl2. Now again from KSP, very important, right? So if you see that what is the minimum pH necessary? pH necessary, you need to calculate the value of OH here. KSP is given to you, right? To form in a this PbCl2 solution. Okay, right. So from here, the amount of PbCl2, Pb is same in Pb2 positive is here also. Pb2 positive is between here also. Okay, that is the common ion. So we can't directly apply the trick here. We have to use proper method here because common ion is there, right? So if you see this here, I'll just firstly rewrite the formula here. So if you see this here, this is Pb, OH twice, that is going to give you Pb2 positive plus OH negative, that is 2. So if you want to write the KSP value that is given to you in the form of ions, right? See, they are giving you, they are saying that they have common ion effect, right? So this is PbCl2, this is going to give you Pb2 positive plus 2Cl negative. This is given to us, that is 0.12. So this will also be 0.12, this will also be 0.12. We need to take care of this 0.12. If here it is 0.12, here also it is going to be 0.12, right? Okay, because that is the common ion between both of these, right? So here it is also 0.12. Here I am taking the concentration as OH negative raised to power the stoichiometry. So KSP is going to be the concentration of Pb2 positive, OH negative, the stoichiometry, right? Pb2 positive concentration you know, OH negative you have to solve. Once you solve the concentration of OH negative, directly you know that if OH negative is equal to 10 raised to power minus 2, take the logarithm here, answer will be, pH will be, what is pH? What is pOH? pH is equal to minus log of OH negative, right? OH negative is 10 raised to power minus 2. This is minus log of 10 raised to power minus 2. So that will become minus 2 into 2, that is 2 log base 10 of 10. That is 1. So answer will be 2. pOH is 2. Then pH is going to be, pH is equals to 14 minus pOH. That is going to be 12. Right? So that is how you will solve the answer for pOH here. That is your C is the correct answer for your pH. Understood? Yes, students. Next question on your screen, everybody. I'm happy you guys are able to answer the questions. The frequency of second line of Balmer series in spectrum of Li2 positive. Li2 positive is the spectrum. They are asking the frequency. The answer is given in Rydberg constant. So you have to use the formula of Rydberg, the frequency they are saying. So we know that 1 by lambda is equals to, this is RHZ square, right? This is 1 by N1 square minus 1 by N2 square, right? Second line of Balmer. Now Lyman-Balmer, that means N1 is equals to 2. Second line, student, what is the value of N2 here? What is the value of N2 in second line? You will add 2 here, so answer will be 4. Okay, third line you will add 3 here, so answer will be 5. That is why they can also give you beta line, right? They can also give you beta line, alpha line, gamma line. That also I have taught you. First line is alpha, second line is beta, third line is alpha. Infinite line is limiting line. Okay, understood? So you just need to put your value here. E is equals to HC. They are asking the frequency here. HC by lambda, right? We know that frequency, if you see, C is equals to mu lambda, right? So frequency is equals to C by lambda. Once you solve the value of 1 by lambda, multiply this with the c here, right? Then directly you will get your answer here, right? So basically this directly you have to solve. RH is given to you. Lithium, acha, this is given you lithium, so Z value is going to be 3 here, right? So for lithium, hydrogen it is 1, helium 2, lithium 3. Beyond lithium they will not give you, it's not applicable, right? So this is N1 is 2, N2 is 4. Directly put the value, solve the answer, and you will get your answer here, right? Okay, just don't forget to multiply it by C. Only then you will get the frequency value if you multiply it by C, right? Next question is, during adiabatic expansion of an ideal gas. Now, if you say adiabatic expansion, that means Q is equals to 0 here. We know that delta U, first law is delta U is equals to Q plus W. That means delta U is equals to W, right? Adiabatic expansion they are saying. So Q is definitely equals to 0. Internal energy we don't know, right? Q is definitely equals to 0 and U is equals to delta U, that is also called as delta E. That is the internal energy here. So answer will be B is the correct answer, right? Okay. Yes, students. I think this is the last question. Yes, this, this is the last question. Let us try to solve this. The question is, the order of stability of carbon ion. Okay, now spam it in the chat box. What I told you, whenever they ask you the order of stabilities, right, what you have to answer according to GOC. What was the trick that I told you, right? What are the different things that you have to see in a carbon ion? Spam it in the chat box. That is BARHI, that is Barhi. So firstly, we will see, is there anything which has back bonding? Is there anything which is electron rich and electron deficient? Anywhere you can see that? No, back bonding is not here, not here, not here, not here. So back bonding is not possible. Aromaticity, I can see that here you have cyclic conjugated and it is planar also. It is having 6 pi electrons. So that means this is aromatic in nature. So because of aromaticity, fourth is going to have maximum. So A and D are eliminated here, right? Next is resonance. Can I see resonance anywhere? I can see resonance here. So that means I don't see resonance anywhere else. So that means after this I'll have third, right? Then between 1 and 2, which is more stable here. Then I'll see the hyperconjugation. But the problem is hyperconjugation is not valid in carbon ion unless and until you have any electron with like any electronegative element like CCl3, right, which fluorine, like any halogen, which can cause minus hyperconjugation, right? So hyperconjugation is also not valid. Let's talk about inductive effect here. If you see inductive effect, this, I think this should be negative. This is negative, right? This is negative. This is CH3. This is negative. Here you are providing electrons from two sides. Here you are providing electrons from only one side. So this will be more unstable because something is already negative. You are providing them electron from inductive effect. Here you are also providing, but less electrons you are providing. So first should be more stable, then second. So answer should be B is the correct answer. Yes, B is the correct answer here. Here you made a mistake, right? Okay, here you made mistake. Remember that in the last part you see the inductive effect here, right? So directly you can see this is primary, this is secondary. You are providing it from two sides. Here you are providing electron from one side. And if something you have positive, it needs electron donating. If you have negative, it needs electron withdrawing, right? This is like stress. You want someone to take out stress from you. You want someone to give you money, right? So negative needs negative, negative needs someone who can withdraw the electrons here. So that is why B is going to be the correct answer here. And that was your last question, students. Let me know in the chat box if you find the questions interesting, right? These are the, see, you can see our last year analysis also. You can see the paper also. You will see that the same questions were repeated. Okay, we have given you the proof as well, right? So right now, after like 2-3 days before, right, there is no benefit, right, to see all the live sessions just in one go. Rather than the benefit is see the live session on that point only, so that when I am solving the question, you are just not following what I am doing, right? You should also solve the question with us, right? That is the benefit of live sessions right now. If you're thinking that, oh my God, live sessions are wasting my time. I know you guys are thinking live sessions are wasting my time. I'll see recorded. See, you're good, right? You're not thinking like, okay, it's not bad to see recorded versions. But if your problem is that you're not able to manage the time, you're not able to solve the question on time, you're not able to see whether all other students are ahead you or what exactly is your point, then rather than seeing the recorded versions, rather than just seeing that, okay, this is the answer that ma'am gave, that sir gave, you should solve the question yourself in the class itself, right? So because if your answer is wrong, it's not like you have to, oh my God, what happened? I am here. We are here to solve the question on that spot only, right? So that will help you, right? So that is very important for you to solve the questions yourself. Now, for those students who are looking for good revision, then students, from 16th till 29th of April, free revise-a-thon is going on, right? Vedantu is providing you that for all the NEET 2025 aspirants. You can see in the description, the link is there. Just get yourself enrolled here and revise everything totally free of cost, right? Then for all those students who want free test series, that is also there. You can check the description box. Free test series is provided to you, right? Then free mock tests are also provided to you. Test series is you can solve it anytime. Mock test means that you have to sit there in the class. So mock test will be provided from 16th to 23rd only. That means only 2 days are left for your mock test, right? Today also I think the mock test is gone. So tomorrow and 23rd, right? These are the 2 days you can give your mock test here as well and there as well, right? Next is free NEET 2025 study buddy for your doubt solving for any query that you have. This is also totally free of cost. You can scan this code right now. Over here, this is the code. You just need to scan it and everything will be provided to you. 24/7 smart study assistant. Instant access to daily study material is provided to you over here, right? Okay, so that's it for today. And I want all of you, whatever you are feeling, I know there are a lot of things going in your head right now, right? I'm feeling anxious, I'm feeling demotivated. Ma'am, I feel like I won't be able to crack the NEET this time. Ma'am, I'm feeling underconfident. Ma'am, my mock test results are constant. They are not, ma'am, they are actually decreasing as the exam is coming near. I know there are so many things that are going on in your head. So the comment section here is your dump box. Okay, the comment section is your dump box. So you just have to dump everything, whatever you are feeling right now. And why I'm asking you to do that? Telling you again and again that it's not only you are feeling this, it's other students also. You will like, ma'am is just okay consoling us. Ma'am doesn't mean what she's saying, right? But when you will see the comments of students, right, you will see every single student is going through the same situation, right? Even the one who was scoring like 600, 650 plus, right now they are going through the same situation. There are just few students who can keep calm in these, 10 days before the NEET exam. So I want you guys to dump everything that you have in the comment section, whatever the negative feelings you are feeling, whatever you are feeling. That can be anything like, ma'am, I am not feeling well, I'm not feeling like studying. That can be regarding your mock test, whatever is there. So that our team, Vedantu NEET English team, Shreyas sir, Gopika ma'am, Baswa sir, and me, your Diksha ma'am, we can go through your comments and we can see what are the problems that you guys are facing. And I'm not talking about the problems related to the subjects. I am talking about the problems that are general problems that you are facing, that may be related to any health issues, that may be related to any anxiety, that may be related to the sleep problem, right? One of my cousin only, he is in NEET exam he is going to give you. He called me yesterday. He was very depressed. That's how I realized that, okay, when I meet students, right, that was my cousin. That's why he was able to tell me that, okay, Didi, I'm not feeling well. My mock test, he was like, I'm taking coaching from last 2 years. but my mock tests are not going beyond 450. I don't know what is the strategy that I need to apply. That's why I made that mock test strategy for you, right? Okay, that was the reason, because when I talk to students, then I got to know what exactly are your problems. So we want to know your problems in the comment section, whatever you're feeling, right? Okay, dump it in the comment section. Okay, yes, that's it for today. I'm waiting for your comments. We are waiting for your comments, and we will surely come up with some solutions regarding whatever you are feeling right now, right? Okay, so that's it for today, and all the best. Now take a rest for 1 hour, and again start revising, and see in today's mock test, right, the questions that our team has solved, the questions that you think you got wrong, right, and the topics that you think that, okay, I was not able to solve question based on that. Revise those topics today itself. Don't procrastinate anything, right? Okay, yes, I'm waiting for your comments in the comment section. Till then, bye bye, take care. See you all in the next session. Bye bye, take care.